see that this surface layer is warmed by as much as 2.5 degrees still in February 2015. The warm anomaly has come onto shore right against Vancouver Island here. Along with the warm anomaly, there's quite a buoyant surface layer, what we'd expect, and a little bit of a loss of oxygen, but not especially. I mean, I'm going to be talking as I go on this about the buoyancy of waters, the density of waters, and oxygen. That's going to be the focus of my talk, and that's why I show these data. Okay, so as I've mentioned, lots of online data. I'm going to be touching on saanich near the end of my talk, but before then, I'm going to be taking a look at Folger data off OMC and lighthouse data, satellite measurements, of course, and as I say, all this data online. The anomaly as it comes to shore, first of all, I take a look at what's going on around the Folger cable site. And here I'm using both the MODIS SST data to give me a bit of a time series and seasonal averages over a broader area. And then at 25 and 95 meters, I'm showing also the Folger temperature data. And so we can see then in the January data point is hidden a bit here, the MODIS January data, but there's distinct warming in January, and by February 2015, there's a warming between 2014 and 2015. Waters are 2.8 degrees warmer in this box off the mouth of Juan de Fuca. So we've really brought quite a warm water mass onshore. Next, these wonderful time series data that you can download off a DFO site show, I think, give a bit of a historical perspective, a time series perspective to this warm anomaly and how it's penetrating the coast. And so we can see here August and February data, and this has been kind of a feature of this warm anomaly that you don't see much in summer. Summer warming kind of masks what's going on, but if you look at winter data especially, then you can pick out something like this. Between 2014 and 2015, we can see 7.5 up to 9 degrees, perhaps, maybe 1.5 degrees warmer waters around Race Rocks. And these are monthly average data. So it appears that the warm water mass is, and of course around Race Rocks, we're looking at a fairly well mixed water column. So the surface data that you get from lighthouses is probably representing most of the water column here. That's certainly true in Harrow. So then I go to DFO. This is again line P cruises that and Marie Robert has started to pick up. Harrow Strait is one of the stations that she routinely samples, and so I can compare now what's happening between February 2014, before warm waters entered the straits, and February 2015. And you can see through the water column now this warming that is maybe, well, yeah, 1.5 degrees, somewhere around there. There's a density difference that's quite striking between the 2 years. In 2015, this warm water mass has affected the density of the surface waters. But interestingly, I think there's not as much of an oxygen signal here. 2015, the warmer waters have not really affected what the oxygen content of the waters that will then penetrate both the Strait of Georgia and Saanich Inlet. You know, there hasn't been a loss of oxygen. If I'm not being clear, interrupt me, please. Looking then at the Strait of Georgia, deep central basin site, again, ONC data. We can see that, what do I want to say here? The low oxygen waters are associated with the warmest waters of the year, which also happen to be the densest waters of the year. And so we look at the seasonal pattern in basin waters in the Strait of Georgia, and warmest waters have not quite the lowest oxygen, but co-occur with the densest waters at depth. Subsequent to the turnover of the basin, or the dense water inflow of the basin, then there is a decline in density over the next year, most of the year, which must be caused by mixing from above. And I think, and some years ago, you did a bit of work showing how strong the inflow was into Georgia, didn't you? On tidal cycles, there were very vigorous. It's not you? I thought, okay, so I thought maybe, anyhow, I suspect then anyhow that the reoxygenation of the Strait of Georgia is dependent on, remember? Yeah, you did too. Yeah, and very strong currents, right, that were coming in through Harrow. Yeah. So anyhow, so there's some energy then on the inflow on neap tides that help then mix down oxygen into the Strait of Georgia, and this is my take on things. It's a little bit different from say the Johannesson paper in 2014, where they actually try to talk about 2 turnovers of the basin in a year, 2 overturns, and I think there's one. I think the Strait of Georgia and Saanich Inlet are very much the same. That you have a late summer intrusion of dense waters, and then you've got mixing through the rest of the year with oxygenated waters coming in over the sill and penetrating to various depths. So one of the things I wanted to point out here was in 2015, how the warm waters, maximum density waters, brought in low oxygen waters, but for some months afterwards, there was no transport of oxygen back into the basin. Oxygen continued to decline for several months to a value that is lower than what Johannesson et al. Suggested was as low as you could get in the Strait of Georgia because of mixing in Harrow Strait. So they modeled water exchange through Harrow into Georgia, and said essentially that even if you had anoxic waters coming from the open ocean being mixed in Harrow Strait and penetrating to the basin, the lowest oxygen you might expect would be about 90 micromole. And because of weak winter resupply of oxygen, we saw lower than that. And this is going to be sort of my theme. We have to pay attention to density as a mechanism for transporting oxygen into both Strait of Georgia and Saanich Inlet. Okay, so this is just recapping what I said. I don't need to dwell on this much. In summer, low oxygen waters actually are transported deep into Georgia, and then the rest of the year you have neap tide injections of oxygen that slowly mix down until you reoxygenate the basin, but decrease the density of the waters in the basin. And so that sets up again the summer overturn of Georgia. Saanich Inlet data. Here's a time series. I wanted to put the time series up to show that the warming at 100 meters that we've seen in the last 2 years of 2.3 degrees between March 2014 and December 2015, as recorded by the Venus site, has taken temperatures about as warm as we've seen in the past 50 years, but has not created a huge warm anomaly at depth. The recovery from cool to warm has been quite strong, and so I think this is evidence, I would say quite clearly, of the warm water mass generated out in the open ocean penetrating through Harrow Strait and into Saanich Inlet at 100 meters. On the bottom, this is again the Venus data that shows oxygen levels and annual cycle. You can kind of see here how weak 2015 oxygenation is, and this is something actually that the Hallam people have been talking about. They've go to the Bamberton site, station SI3, and they saw, I think, no basin overturn in 2015. Is that correct? And a buildup of hydrogen sulfide and so on. So this is the trick of interpreting what's going on at a mid-depth in Saanich. You have to understand what's happening in the basin below. And so I showed then, oops. Next thing I'm going to do is just overlay the oxygen data from the node and show you what 2015 looks like compared to all the previous years. And you can see then that there's this extended period early in the year through January, February, with very low oxygen, some spikes, but overall, this warm water mass must be weakening circulation in Saanich and reducing oxygen supply. Profiles, the profiler is out now, down the arrow point. And so there's 2 years of profiler data, 2015 and then 2016 in February, that show quite low oxygen. Just this 95-meter depth is just exactly right where we see the oxygen minimum zone building up to recently. So I went to the UBC data to show what 2014 looked like in February, and you can see that this area is a very active one. And I would suggest then that what we're seeing in 2015 and 16 is directly a product of the blob. So Saanich Inlet, I forgot to say, what I did want to point out here was basin warming was quite slight, 0.3 degrees perhaps, between 2014 and 15 and then 2016. So the warmer waters would have come in summer 2015 in basin overturn. It's a weak signal, and I don't, understanding where waters come from to get into basins, it's not surprising to me that the warm patch out in the ocean didn't affect the basin waters that much, because a very large component of those basin waters is the upwelled waters from the California undercurrent that enter Juan de Fuca, are weakly mixed in Harrow Strait before entering Saanich. So that's the typical summer circulation, and winter then we allow this anoxic basin to survive because the inflow waters are fresh and don't penetrate very deeply. And there's been a little bit of talk. I wanted to make sure we remembered that the anoxic boundary sloped up the inlet. This was shown by Dick Herlenbo many years ago, and Roberta talked about seeing that too in their data when they headed further down inlet. So as I've been mentioning, we really have to pay attention to the density of these waters that are coming into these inlets, the changes year by year, and how that controls the development of hypoxia in our coastal waters. So taking a broader look then, how might climate change increase hypoxia in BC waters and Saanich Inlet? I'd say the recent warming event is a good example of how decreasing the density of the mixed layer can result in much weaker oxygen transport in winter into Georgia and into Saanich Inlet. And this is something we should anticipate as occurring in the future. Summer upwelling appears to be strengthening. Okay, so there's some papers out that have come out recently talking about the poleward enhancement of upwelling because of the shifts shifts poleward in wind patterns. And so there's some modeling effort, and I've looked at coastal data, some evidence in upwelling records that shows that upwelling seems to be increasing along the BC coast, especially further north at the north end of Vancouver Island. I can see that quite nicely. I took a look at that because we're trying to understand how climate might impact sponge reefs. And if we end up with stronger upwelling, we're certainly going to get more low oxygen water onto the shelf. And I think in Queen Charlotte Sound, Hecate Strait, that is a serious issue for the deeper sponge communities. It's got to also have some impact on the waters we're looking at in the southern part of BC. Another issue that we have to pay attention to, I think, is as we shift from a spring freshet to more rainfall in winter, less snowpack, more rainfall in winter, how is the change in the Fraser River discharge going to affect the density of waters flowing into the straits? I'm not going to think about that too hard, except to say that's something we do need to pay attention to when we're trying to model how inlets get oxygenated. And then finally, I believe that microbial processes should be boosted as we warm basin waters. And so I think those are the thoughts I have on how going from the blob to understanding what warming of oceanic waters, how that might impact coastal waters. I think I always head towards hypoxia and thinking that is the big issue for a lot of our coastal waters. And so those are sort of my summary thoughts. I'll leave it there.
Questions? Jody.
Frank, I was a little confused about the dichotomy between it just being heating in the surface layers and the water is supposed to be flowing out of this rainwater.
And that's right. And that's why you don't get such a strong surface signal, I'd say. But there's enough mixing in Wanda that heat gets down into the deeper waters. Not completely, but some component. I think that's why you see in Saanich Inlet, for example, you see the basin water is only warming by 0.3 degrees because you haven't really managed to mix that heat down to the deep waters very well. So that's summer. So in winter, I would say because of coastal downwelling, the warmth, the anomalous warmth can come throughout the water column into Wanda Puka. And that's probably why we're seeing a strong warming signal at 100 meters in Saanich, because that's a product of what's being imported from the coast of Vancouver Island during a downwelling season.
Yeah, I would suspect it's the downwelling
that brings in the heat. Yeah. So thank you for mentioning that. I'd agree. Upwelling doesn't bring in much heat. Only the bit that you can mix in from surface waters. The downwelling system is probably where you're transporting.
Also suggested there was anomalous southerly winds, so
True. Good point. Yes. Yeah. I'd agree with that. Yeah.
I think those two CPE casts in Harrow were really quite telling. I mean, there's clearly a lot of more heat coming in.
And it's mixed.
I don't know. Carol's rate changes.
Maybe the timing of those casts would be good. Okay, you have, if those casts were a spring or noon, they go that kind of range. You take whenever line peak cruises.
That's right. So it's just opportunistic. Absolutely.
But she's got to, if you look through the history of those casts in Harrow, you'll get a feel for how variable it is and whether a 1 degree over the entire water column. When I tried to do some heat and a slide I put together, I brought back from Seattle actually, was looking at an Argo compiled data set out in a big area of the Northeast Pacific, Gulf of Alaska. And it's a time series averaged over a huge area, showing where Frank showed the line P data showing it's very much at the surface with a few sort of patchy eddies, if you like, underneath. This time series from the Gulf of Alaska shows, or more or less, sorry, not even the Gulf, more or less right west of Vancouver Island, shows this surface signature gradually deepening over the last 2 years. I mean, it starts off the first year at 100 meters, but in the last year and a half, it's penetrated down to 150, 200 meters. It's more of a time series. It's from our book float.
Yeah, do you see a seasonal difference there? Would you see downwelling, upwelling in that?
No, this is too far offshore.
It is, okay.
It's
That's
way
further up.
outside of the upcoming.
Yeah. So it's mixing down.
More questions?
No, that's good, thank you.
Yeah, no, thanks.
Yeah, thanks.
So next up, Steve is going to focus in on this apparatus that we've installed since we had the last saanich in the workshop in 2012. We now have installed the void profiling platform in saanich inlet. It's just north of S3 and south of S4 stations in saanich inlet, located in about 210 meters of water. And Steve's going to show us some of the data from there. We had some cable issues this past year. We replaced the whole cable, and then we replaced the cable and then the terminations. But it's up and running. So, Steve.
So my name's Steve Mahaley. I'm a staff scientist with Ocean Networks Canada, as Fabio was just is, and Akash is, and work for Richard Duke. So I'm going to talk about time series water column measurements in saanich inlet and basically introduce the void profile system. This will probably, some of the data here will illustrate the fact that if you single casts of CTD casts are often cannot represent what's happening because they change in a very sort of rapid sort of way, and you need to have this longer time series of regular casts. So it's an interesting sort of segue from the last talk where we saw great changes in Harrow Strait. It's hard to determine whether that was a change due to a aliasing issue or it was actually a long-term change in the conditions in Harrow Strait. So I gave this talk or a bulk portion of this talk at these are my co-authors, but they basically have not seen this talk, and any errors and wild speculations are strictly due to me, so I just thought I'd leave their names on. So this is the situation in the Northeast Pacific now. Those green dots are void profile systems or vertical profilers. Just this last year, we managed to get a group of them off of the Ocean Observing Initiative. There's 5 off the coast of Oregon, and 1 out the Station Pub. So basically, this type of measurements are going to be much more prevalent in the literature, I think. It'll be interesting see how that changes our understandings. So our situation here is basically in saanich inlet. I'm sure you all recognize this. We're about 3 kilometers south of our void profiler is about 3 kilometers south of the instrument platform, and it looks like this feature here. Bad operating these kind of things. So it's about a 30-foot across surface platform. It's an old CPEX buoy, if people remember that experiment. And here it's leaving the seaplane dock and heading off to its position over there. It's basically right in the middle of the tallway at the beginning of the deep basin, so it's about 200 meters of water. And it's narrow, might be representative of what's happening throughout the width of the inlet. Whereas if you out here, there seems to be quite a bit of cross-channel very quickly. Okay, here it's made of 2 components basically. The float and the profiling package, and it's moored in this 3-point system. And here's the float. Basically, the float, as I mentioned before, has quite a bit of room to do stuff on it, so there's a bit of real estate there. It currently carries a MET station, temperature and pressure, relative humidity, wind speed direction, as well as a downward-looking 200 kHz echo center. The profiling package, which profiles the entire water column, basically is stowed at depth to minimize fouling, and it rises to the surface and measures basically CTD stuff, CTD oxygen sensors, both a fast response Rinco and SB43, fluorescence with a wet labs FLNTU and mass scatter turbidity, and also a path length transversometer.
How deep is the instrument package when it's at the bottom of the float?
When it's at, how high is it? I actually,
How far below the water?
Your shallowest data point is about 3.5 meters. Yeah.
it is, but when we actually look at the data, we tend to, that gets so noisy there, we tend to remove a lot of that data. And so, yeah, I can't remember what my first data point is. Basically, it's deeper than that. It's very noisy up there. I don't know how much the flow itself affects the data. Those are open questions. But so we're recovering the BPS this spring, and it's going to have some instruments we'll be able to move off the BPS and put on to BPS. Probably the PCO2 sensor and nitrate sensor. So we'll have those additional instruments, and basically we have this view instrument that Cash is working diligently to procure. It's an underwater vision profiler. It's based on this instrument, which is a laboratory instrument, basically using a scanner with red light. You put your sample in the scanner, and it scans the animals in the fluid, and there's some automatic identification software that figures out what's there. In this case, basically it has about a 20 centimeter square area where it illuminates with red light, and then there's a camera on top of it that takes a picture of those things about 20 times a second, and you can get an idea of possibly predator-prey relations and stuff like that. Here's some samples of what it puts out. Basically, I think there's automatic identification for a lot of these things, and we should be able to it should be a really valuable sort of addition to what we're getting from the ZAP that's looking down here. So we can sort of ground truth the ZAP data with what this profiling system is measuring. And there'll be some avoidance issues, obviously, but it'll give us a better idea of what the abundances are of different animals. Anyhow, so basically, the data I'm going to be looking at was collected from 2014, beginning around July, where it was manual operated for 2 cycles a day, so about December of 2014, where it got automated. There's a lot of dedication to do those 2 cycles a day for about 6 months. And then it was automated till February, and where it got the ground fault. And we've been slow to repair it, but it's back in action now since January of 18th. And we've got it running at 4 cycles a day. Basically, at noon, it comes to the surface. It takes about half an hour to get up to the surface, which I noted is almost exactly the same timing as zooplankton, so it's an interesting coincidence. And then it goes back down to the seafloor to about 10 to 1. So this happens at midnight and noon and 6 and 6, basically. But this could be modified in any type of way with anything from anybody in here to possibly be a better sampling arrangement. Currently, we're trying to limit it to four cycles a day to see how long we can get this thing to last. Because it'd be really good to have a continuous year-long cycle anyway before we have to go in there, replace the tether, which tends to wear out due to sleep. Okay, so this is the now famous.
Good question. Does it sample on the way down or the way up?
It samples both ways. No.
So there'll be a wait.
Yes. So it's samples, but
It samples all the time, but.
Yeah, it's on all the time, but we take it down as the, idea of capacity.
And I actually can't remember how I sort of, I kept the data over an 8-month period, and I don't remember how I sort of got rid of the up and down pass. I think I just mashed it all together at this point. But anyways, here's Ann's paper, I guess, and Dario's and Frank's paper, basically a plot from it, and most of the conclusions here or the thoughts here come from that paper anyway. So Saanich Inlet, probably as you might have heard in previous talks, is a quite peculiar fjord. It's north facing. It's 24 kilometers long, not particularly deep, just greater than 200 meters, and it's got a very wide and broad and deep sill, basically encompassing this region up here and here, all through that whole area. So it's not straight up fjord. The processes that control fjords, physical processes, don't really exist. Basically, it has very little estuarine input in the head, and probably more from Shawnee Creek, and often is considered a reverse estuary where the fresh water is out here, flowing into the estuary, and then pushing water bottom. That's not the regular estuarine circulation. Also, the fact that it's northward facing like this, and sort of surrounded by this landmass here, is protected from the strong easterly, southwesterly winds in the winter. And it doesn't have any katabatic kind of winds, there's no snow-capped peaks on either side of it. And the daily sea breeze and land breeze effect doesn't really occur the same way it does in other fjords, because most of it is happening up in the Fraser River Valley. So it's weakly mixed. The bottom waters, of course, which makes it really interesting, are usually devoid of oxygen. So here's this probably has been shown before. This is our 10 years out time series of water properties at the Venus instrument platform. Basically, we're going to look at a period here that matches nicely with the period that was looked at before by Karen Manning and others. So I just found out it was an undergraduate student. That was an excellent paper that she managed to get together. And basically, it shows that both these years were anomalous years. Their year was the El Nino, and ours was this precursor to, was basically a blob year. You can see the effects of the actual water properties here are quite similar to the El Nino year. You can sort of see that the density is very, very evident. It's a different, that there's effects from the blob, or the conditions that cause the blob are affecting the conditions it's actually the same way. And one of the main finds is that we basically show that what Manning et al. Found in 2009, or whenever the paper was published, are repeated in our. Okay, so here's some plots of the BPS data from July till February. Basically, we have salinity, temperature, and density. And the things to pick out of here is this sort of fortnightly, monthly kind of cycling happening in all the structures, all the plots here, and the fact there's a very strong change from summer to winter modes. The density does remain more consistent throughout the year, because there's basically it's controlled mostly by salinity. That doesn't change as much, but any kind of freshwater inputs are balanced by cooling of the surface water. So you get a very consistent density profile. So I'm going to basically use oxygen as a proxy for renewal. Basically, the inlet itself is just depleting oxygen by respiration, and we expect that the oxygenated water is coming in outside of the actual inlet from different mixing regions, and we try to trace the flow of the water. So I've broken up into 4 areas that they're kind of distinct. It's pretty obviously distinct here at the surface, down to about 30 meters or so, a mid-depth region, a deep region, and this very bottom region. Now, at the surface, we see we've got both what looks like monthly and fortnightly pulses in the end of the summer here, some cessation of events here, and then a steady fortnightly kind of cycling happening during the winter months that we've observed. Mid-depth, similar sort of pulses happening, and a out-of-phase fortnightly pulsing happening during the winter months. And the deep, you can't really see that well, but this plot, there's some input of newer water during the fall period. And the bottom stays the same throughout this particular period. So if we want to try to understand the phasing and timing stuff, I'm just using basically pressure from the Venus instrument platform, but we know that the mixing is occurring that's generating this new renewable waters that's happening outside of the inlet and at places as far away as Harrow Strait or even Victoria Sill, and there's a propagation time that can mess up our timing quite a bit. I don't know where the water originates from these. But we might be able to use this timing as a way to find out where it comes from. So, but I basically just drew these lines on here at the peak or the nadir of the Neaton cycle. And basically you see that in the summer months, we've got both monthly here cycling and fortnightly cycling, whereas as we head towards the winter months, we get a regular, what looks to be during the peak or the spring tides, and at the surface layer and the mid layer, we have that at the actual Neaton tides. So they're out of phase, quickly out of phase with each other. Also, we can see these deep intrusions correspond to sometimes a lowering of oxygen. And this is like, I think, another plot was shown where it's dangerous to be at this 100-meter depth right at the oxygen level. So you push, the theory is you push up basically deoxygenated water by the influx of this new more oxygenated water, and that's why you can get some out of phase stuff, and some of the stuff is actually in phase. So now if we consider the standard sort of de facto kind of description of saanich inlet as a reverse estuary, and you try to look at the data that we have here and try to put a slightly different model onto it. Basically, we just concentrate now on winter months. We see a pulse in the surface and then a pulse at the deeper layer. So what I'm proposing as a model is that during spring tides, water is in some manner propagated into the inlet to the position where the oil profiler is and pushes deoxygenated water out in this deeper layer. And that alternates on a weekly basis, basically, with now water propagating at deeper depths and pushing the water out in the surface layer. So we have this back and forth reversing estuary circulation. And that's the law of regulation here, basically. So if we look at the summer months here, where at the beginning we have the same sort of periods of strong oxygenated water coming in during the spring tides, but we also see that there is as we head into the fall time, we have these deep intrusions of oxygenated water, and that sort of appears to reverse the whole circulation here. My theory is that this stuff comes in, basically pushing the surface waters out, and we have more of a regular estuary circulation, even though it's not forced by a river, it's at the head, but actually by the intrusion. Again, another speculative.
To comment on that, I suspect because your oxygen levels are so high in summer, that's probably part of productivity rather than transport of oxygen.
Well, the range of it, but the fact that it changes on this fortnightly cycle.
And it's not a transport of oxygen, it's all transport.
Right. Yeah. Unfortunately, we do have fluorophosphorescence, but I didn't look at that data. It's basically, it's only the surface. I should have looked at it because then we could make that connection a lot clearer that oxygen is.
And just looking at oxygen saturation on the readily too, that's the calculation we did.
Okay, so that's my little model, and this is basically what we have coming out now for the last month or so. It's not quite a month, since January 18th. And this is data available off the actual website. I just picked it up yesterday. I didn't even know it existed. It's pretty good and stuck on here with the pressure at the Venus instrument platform. And we see basically at the spring tide region, we have this sort of influx near the surface of more oxygenated water. So kind of following the same sort of thing that we were doing in the longer period. And then in the sort of neap tide, we get possibly a deeper influx of oxygen here. But that's sort of the point I'm trying to make with this is that the previous plots missed this very important region between the end of, or the beginning of February to the beginning of July, where you will see whether that whole cycle will now connect back to itself or whether there's an evolving sort of multi-year changes occurring. If you look at the previous plots there, basically, yeah, not so much there, but here, you don't see any sort of, you don't see how this end part here is going to attach back to here. So something really big happening through this region here, the spring region, probably some renewals as was shown in the camera. Anyways, that's basically about it. There's a summary of what I've said, basically that the BPS location, at least at the BPS location, the model that there's a reversing estuarine circulation happening is quite can be used to explain the structure during the winter months, and possibly the structure in the summer months returns into, or the fall months returns into a regular estuarine circulation due to the renewal of deep waters. That's it. And here's the actual, a very clean picture of the downbroken echo sounder. And this is what I mean by noisy data. It's only penetrating down to about 30-meter depth here. And I'm not sure whether that.
So that's what you're seeing there is the time variable gain is saturated. So it's just,
yes, it's
ramping up and hitting the ceiling long before it needs to. So that's what May was talking about, is either calibrating or changing the time variable, removing or correcting for an overzealous hardware time variable gain to take out that background gradient, which is not real so much as it's right there, but it has a compensation hardware in there that approximates the time variable gain, and you need to sort of remove that.
But there would be a limit to that depending on
yes.
SNR. So the question is whether we can fix that to go down.
Whether there's any signal left in there right now.
And the other, can't remember what time it is, but it could be large reflectors or backscatters that are causing us to not be able to do that. Yeah.
Okay, thank you. Okay, any questions for Steve or for, in particular, this idea that the void profiler is out there, it's a platform at the surface to hang gear on with a med station. It's got an echo sounder looking off at an angle. It's got this profiling package that goes down at the moment 4 times a day. There are still ports available for it. We've got a number of instruments lying in the queue. As you pointed out, there's a an imaging system we're in the process of purchasing to get on there. But it's also going to accommodate additional instruments. Yesterday, Steve Hallam talked about his environmental sample processor, which is a genomic probe device where you can take a sample, filter it into a puck, and it'll actually do a local probe test to see if the community dynamic or community composition in situ. That is a big instrument. We're looking to get a pressure can for that, and then if that would potentially go on the void profiler, it would be a very massive kit to add there, but that's the goal was originally to put the ESP on the void profiler. If you have an idea for an instrument or sampling or a sensor that you think needs to be on there to support your research, make sure you contact myself, Cash, or Steve. We can talk about it now. There is a transfer somewhere, yes.
Encourage you to keep it. Have to clean it.
You have to clean it. Or that's a straightforward.
How often do we have a sort of schedule now? Right now, the engineers are very busy, but.
Ideally, every once a month, perhaps during the production time of year. What about mass?
What? On that spec?
Yeah.
We've been talking about that ever since the beginning of Venus. And, we don't have the funds to buy one, and for a while there, they were only renting them. They wouldn't even sell them. I think some of the manufacturers, at the moment, there's no plans, but if somebody wants to buy one and put it on there, that could be considered. There you go. Okay. All right. So next up, before the lunch break, it's going to set us up for this afternoon is Roberta Ham, and her title is right there: Interactions of Nitrogen Cycling and Water Mass Renewal in saanich.
Thanks, Richard. I'm Roberta Hahn, and I want to talk to you today about two projects that we've done already in Saanich Inlet, both focusing on nitrogen cycling and the interaction of that with water mass renewal, which you've already heard quite a bit about today. And then at the end, I want to talk to you about a new experiment that we want to do in Saanich Inlet this coming year. Okay, so yesterday we heard a little bit about the nitrogen cycle and its various complexities, and I want to simplify that down. Instead of looking at it from this kind of angle, we're just going to look at it like this. Okay, so any bioavailable nitrogen, I'm just going to make that all one pool. So that includes all your nutrients like nitrate, nitrite, ammonia. And then thinking about that in terms of its interaction with nitrogen gas, which is generally not bioavailable. So what controls how big this pool is here? We have nitrogen fixation. There's some bugs that can take this N2 and they can make it into ammonia. And then we have processes that take these nutrients and transform them back into N2. I'm going to call all those processes denitrification for the purposes of this talk, but I mean all those things: anammox, canonical denitrification, anything that takes bioavailable nitrogen and makes N2 gas out of it. And we're really interested in this balance because the size of this pool, bioavailable nitrogen, controls essentially the productivity possible in the ocean. Okay, so this process we've already heard about, denitrification, happens in low or no oxygen waters. And we've seen a couple plots like this already in the symposium. This one has to be oxygen at 200 meters. So you can see these low oxygen areas here in the eastern tropical North and South Pacific and in the Arabian Sea. And these are areas where we see denitrification happening in the open ocean in the water column. And the point I really want to make is that these areas are really controlled by a combination of biological processes and physical circulation. Right? So over here, you've got upwelling zones, so there's quite a lot of productivity in this area, but these are also shadow zones in the gyre circulation. So if you picture the North Pacific gyre, it's coming around like this, and so these are very poorly flushed areas. And so that's kind of but obviously the way that circulation works in Saanich Inlet is quite different from the way it works in the open ocean here, but still those same kind of factors come into play. All right, so a lot of times people look at denitrification by looking at nutrients, by comparing nitrogen and phosphorus. And in our lab, we go about it in a different way. We're looking more at the product of this denitrification, at the N2 that's produced. And what I'm going to show you today is nitrogen-argon ratio. So that's what we measure on the mass spec, but also the ratio with argon takes out some of the physical effects on nitrogen, so we get a more purely biological signal. And what I'm going to plot for you is the ratio that we measure, or the ratio at equilibrium, and -1 times 100 to get them in kind of nice-looking units here. So here's a couple examples from Al Duvall's lab at University of Washington. This is the Arabian Sea. So you can see these high nitrogen values in the denitrifying zone there. And also this one for the Eastern Tropical South Pacific. Again, high nitrogen values. Okay, so we did 2 studies. One was looking at discrete samples that we took, and that was at a station that was pretty near the ONC node, but in the middle of the basin instead. And this was in 2008-09, that's the Manning paper that got mentioned already. And then we also did a study using in situ sensors to get at nitrogen values at the Venus node. Okay, so first turning to this 2008-09 time period, this is what the oxygen looked like during our cruises. And so we saw this kind of mid-depth or bottom, but not, deep, but not all the way to the bottom renewal happening here in May, April, May. So we saw a shallower oxic-anoxic interface that deepened. Then that stayed quite stable for about 4 months. And then we had several deep water renewal events come in fall of 2008. This was quite a strong renewal year and really increased the oxygen here. That oxygen disappears by December, and we have a return of anoxic waters. Okay, here's Kara. She collected the sample. She really led this project. She was an undergrad that worked with me. Also, I want to acknowledge Annie Bourbonnais, who was a student at Kidd University who worked with us on this. So this is what our sample flasks look like. They take about an hour to analyze each one, so we have to cryogenically process them to get rid of water vapor and carbon dioxide. We run them here on the mass spec. So this is more kind of small amount of data, but we think that we have quite high precision in it. Okay, so let's have a look at it. Here's our first profile. This is April of 2008, the nitrogen-argon ratio. So down here in the deep waters, which are anoxic, we see very high values. This is from all the denitrification that's happened. So this water is completely lacking in nitrate at this time period. This amount that we see is the highest, by the way, that we've ever measured in the ocean. So it's almost twice as high as what's seen in the Arabian Sea or the Black Sea for this value. Okay, so then we have that mid-depth sort of deep renewal, but not all the way to the bottom come through. And if we compare that with these next profiles from the summer, you see that we've lost some of the nitrogen here. There's a bit of flushing. This is where the flushing occurred. And we gain a little bit down here in the deep. We think that there's still active denitrification happening down here or near the boundary due to mixing, bringing nitrate down to that depth. Okay, so then between August and October, we have the big water mass renewals that are flushing this water out. And the nitrogen drops quite significantly here. We see higher nitrogen higher in the water column, so this nitrogen N2 was pushed upward. And we can calculate here that based on these values, we think about 45% of the bottom waters in this year were replaced with new waters coming in. Okay, and then the oxygen disappears by December, so denitrification can start again. The new water that came in brought nitrate with it, so there's a new substrate for that reaction. And we see an increase in N2 gas here in December, and then more in February, and April's not on here, but it was again a little bit more than that. So the cycle sort of comes back to a similar level that it was at before. One of the things that Kara really looked at was thinking about the timing of these renewal events. And so we used the ONC node data for this, and I want to thank Jody Climack and Richard Dewey for their help in understanding this. This is potential density at that 95-meter node in Pat Day there. And you can see these step-like increases whenever these water mass renewals happen. And the April one is a much bigger increase, and I think that's because the water is really coming in closer to that depth. Whereas the deep water renewals, we have a higher density of water coming in that's coming down deeper in the inlet. This is filtered, this is tidal current speed. We're using active paths here for our tidal current observations. So here we see that the time period where we start to see an increase in density at Venus is really correlated with the lowest mean in these tidal currents. And down here we see an upwelling index as well as the Fraser River outflow for this year. Okay, so the idea that I think has been talked about already is that during these very weak tidal currents, You can have dense water that's upwelling on the coast, essentially sneak all the way and make it to Saanich Inlet without being mixed away. And one thing I really want to emphasize, and it's really important to look at the current speed and not the tidal height here, because of the superposition of diurnal and semi-diurnal tides in this area. The Nepean current speed is not that well correlated with the Nepean tidal height. Mistake that I almost made in this paper, but someone saved me. Okay, so now I want to turn to this other time series that we did in 2011 to 2014. And here we're looking at it. This is just focused exclusively on the node at 95 meters now. Plotting potential density over that time period here, and oxygen here. And this has been talked about already, but I want to emphasize again that we, at this level, we kind of see almost the inverse in the oxygen cycle that we see in the deep water. So during the fall, when we have oxygenated waters potentially coming into the deep water, that's when we see the lowest oxygen at mid-depth here. Let's see. And when we have the highest values at mid-depth, that's when we have the sort of depth of the anoxia in the deepest waters. Okay. And this inverse is pretty well understood already. There's a number of nice figures where people have looked at oxygen time series in the inlet. This is one. A couple of previous papers have some other ones where you see the deep water renewal here. So here's one here pushing this anoxic water up into the shallower depths, where a place like the Venus node can then observe it there. Okay. So in this study, we also looked at nitrogen, but not by our discrete mass spectrometry methods. We instead used a device that's on this Venus node called a gas tension device. And the way it works is kind of a very elegant idea. There's just a headspace here and a membrane. And gases can diffuse across the membrane and liquid water can't. And there's a very accurate pressure sensor in here. So what we're measuring with this thing is the total partial pressure of gases that are dissolved in the water. So that includes the partial pressure of nitrogen, oxygen, water vapor, argon, CO2, other minor gases. And since we have an oxygen sensor paired with it, we can measure the oxygen. We're measuring this partial pressure, the total gas tension. And we can pretty well estimate from temperature and salinity the minor gases, water vapor, argon, etc. So from this, we can calculate how much nitrogen is there. All right, but here's what we see. So it's the same, part of the same plot I showed you before. Here's your oxygen up here, here's your potential density here, and now I'm showing nitrogen excess. This is a little bit different from the nitrogen argon in percent that I showed you before. This is a concentration we think that is in excess of equilibrium. So some of the excess is probably physical, bubble processes and other things can raise the apparent nitrogen excess, and some is biological. And so what do we see here in the sort of overall look? So this time when oxygen is going down and potential density is going up, we see nitrogen going up too. So that makes sense with these fall renewals coming in underneath the water at 95 meters and pushing the high nitrogen water up. So this water can't be, this nitrogen can't be produced here because the water in this time period has enough oxygen to prevent denitrification. And then we have the spring sort of flushing that's happening at the Venus and shallower. So this is not the same as the mid-depth renewal that I talked about before. This brings that winter water in with higher oxygen and presumably lower nitrogen because it's coming from an area where there's not denitrification. Okay, I really want to focus on these renewal time periods. So I'm going to blow up just those two periods for these years. Here they are. Okay, so this is 2012 on the left side. We've got 2013 on the right side here, and potential density on the bottom. So you can see again these step-like increases when a renewal happens in the fall. And I've highlighted that with these gray bars here, the time period that we think that is happening. Look at the oxygen. You see, there's a general decrease here. That's respiration happening during this period. And when these renewals happen, you see sort of a faster decrease, right? It's kind of like a dip that happens there during the renewal period. And that's the same time we see nitrogen go up. And then right after that, when density has stopped changing, oxygen spikes upward again, and nitrogen comes down, but not as far down as it was before. So it sort of partially recovers. So it's kind of like a rebound effect that's happening after these renewals here. So thinking about why that would happen, we've already kind of talked about this deep water renewal mechanism. This is really how I thought about it before I was doing this work. You know, when there's dense water, it can come over the sill, it sneaks down here, and it pushes this low oxygen, high nitrogen water up, and we can observe it then at the Venus node at 95 meters. We think about that from a sort of turn us around here. Now I'm looking at a cross section across the inlet, still at the latitude of the Venus node. So Venus is sitting here in Pat Bay. Again, you know, I really thought about this as deep water coming in at the very bottom and pushing this water up here. But what's really necessary, let me go back a second. What's really necessary for something like this to happen, this rebound effect, is you have to be mixing with waters that have the same density, like the waters at Venus have to mix with water somewhere else with the same density, because density doesn't change, but with lower nitrogen and higher oxygen. And so what we thought must be happening is that instead of water just really coming down in at one density down at the bottom, we really have waters coming in at many different densities all along, and they're going to hug the western side because of the Coriolis force. So if they're going south, they're going to hug the right-hand side, and so they're hugging this western side of the inlet here. And so we have new waters being in place sort of all the way along here. And the first effect you see is still that uplift. But then a couple days after that uplift has stopped, there's a ability for waters over here on the western side and waters over here on the eastern side to laterally mix with each other and create that rebound effect. And we were able to actually go out and observe this a little bit. I've looked through a lot of the historical data trying to find cross-inlet surveys, and people like to just go right down the hallway taking their station. So we didn't find too much. We were able to actually take some ourselves. So this is just some CTD data at 5 stations right across the inlet here. The star is the node. And we've got 2 time periods, about a week apart. And here is the potential density during that time period. So the first one was before a water mass renewal. And the second one is sort of just right at the end of the density change before the big rebound in oxygen. This is oxygen down here in red. Okay, so what you see is before the renewal, all the profiles across here, so they're color-coded according to the map here, look pretty similar, right, on average. But right after this renewal, what do we see? We see waters on the eastern side having quite low oxygen. These are oxygen concentrations starting with depth. Here's a central water, the central station here in the green, and on the west, quite a bit of oxygen. Okay, so we do see evidence here for, right at the end of this renewal, higher oxygen on the western than on the eastern side that might be able to drive this increase in oxygen, but stable, no change in density after a renewal event. Okay, so just to come back here, that's what we think is happening when we see these increases of oxygen after each of these renewal events. And the nitrogen comes back down, but not all the way back down. So it's mixing with this new water that has low nitrogen in it. In this event over here, in this year over here, we see a couple of really quite large increases in oxygen after the renewal, and not as much happening in nitrogen. And looking at some of the CTD data from this time period, it looks like 2012 is a much bigger renewal year. We see oxygen right down to the bottom, whereas 2013, there is not as much oxygen coming into the inlet in that year. Okay, so from our studies, you know, these oxygen deficient regions where denitrification can happen are really being driven by a complex interplay, not just of the high biological productivity driving high respiration, but the circulation dynamics are really important to creating that low oxygen environment. With the discrete nitrogen measurements that we've made, we were able to look at flushing amounts. So I've mentioned we saw like 45% flushing. We were also able to estimate total denitrification amounts and sort of broad scale rate estimates over month-long timescales. One can predict pretty well the potential renewal time, so not whether it will renew, but whether it might. From these tidal current speeds, active pass works pretty well for that. And the in situ gas measurements that we made in the ONC node really helped us to see this other mechanism that we hadn't thought about before, the way that there's kind of a rebound after a deep water renewal event. And I want to tell you about some future work. And that is that we want to do an experiment in this coming year. The idea would be to do a time series from about September 2016, when we're hoping that the inlet would have June 2017, when it should be fully anoxic and sulfidic again. We're looking at approximately a 2-week frequency. We'd like to go a little bit more often than monthly in order to capture some of the transitions, but there's a lot of samples we're going to take, and we can't do a lot faster than that. We're thinking to focus on the site near the profiling mooring, and again near where UBC has their time series. We really like to use the profiling mooring to kind of fill in the time periods when we're not there. So having it right there is nice. We also have an additional 2 days of sediment sampling, probably freeze pouring. I'm not too involved with that part of it. Vera Pospolova is heading that up. So if you're interested in sediments, you should talk to her. So we've talked a lot so far about oxygen in the inlet, about nitrate in the inlet, some about sulfur and methane. There's a lot of metal reactions that are happening too at different oxygen, low oxygen states. So this is kind of an idealized idea, and this is in pore waters and sediment. You might see oxygen decreasing, nitrate increasing, manganese and iron reduced compounds showing up from manganese oxide reduction, iron oxide reduction, and then sort of sulfate and ammonia. And I think one thing that we don't understand very well is how these different reactions overlap with each other. At what points do some of them turn on or turn off? When do they become possible for the microbes to do? And this kind of stuff has been mainly studied kind of in a steady state sense, right? We take a tour through the sediments and we look at these different levels of things going down into the sediments, or we look at some of the stable places like the Black Sea and that sort of thing. But I think Saanich Inlet is a real opportunity for us to look at these reactions in a more temporal way. And also say that I've talked about the importance of denitrification to the nutrient supply, but redox states are also important. Carbon burial, metal solubility, so there's a lot of reasons that we might want to understand these reactions better. Okay, so a lot of these redox couples have been measured in Saanich Inlet. I don't really need you to read all that just to get kind of a sense of, wow, there's been a lot of work. But most of it's all been in the summer. It's all of July, August, a lot of the work that the people from the University of Washington did. So almost every redox couple you can think of, someone's measured it in Saanich Inlet, but they probably were only there in July or August. They haven't really looked at how that changes over time. This is just sort of the early part of it. I have another couple pages of this, but I was going to show you that. So I think that Saanich is a real opportunity, again, to capture this transition. We can look at when these redox couples start to really turn on, when we start to see big changes for different ones over different periods, because we have this regular or semi-regular oxygenation to sulfidic kind of transition. Of course, the data shown just yesterday suggests that that transition is not as regular as we perhaps once thought. So I'm really hoping for renewal this year. Okay, so here are intended measurements so far. This is a coursework that we're going to work with Jay Cullen here at UDIC. He's a big part of this, looking at iron speciation as well as total iron, copper, and zinc. Al Mucci at McGill is doing some other metals, cadmium, silver, lead. I'm going to be doing my N2 gas, but also manganese speciation, which should be an adventure for my lab. We're working with Karen Castiati at Stanford to do isotopes of nitrite, nitrate, and N2O, which will be very interesting, I think. Michael Whitaker is going to look at some methane and methane isotopes. Lisa Miller, working on some of the carbon parameters. Deanna Barella with productivity, nutrients, chlorophyll, etc. John Dower, secondary production. And these are all the PI names, but really there's graduate students and things working on these. And Kim Juniper looking at dark CO2 fixation, rates for some of the nitrogen and some metagenomic and other molecular kinds of things, and maybe some more stuff out there. We're really looking for collaborators on this. Okay, intended continuous measurements. So of course the central node here at 95 meters is still very important to us, and I think there's certainly plans that will keep going. We're excited to see that the profiling mooring is once again doing its profiles, because we think that'll be really important in thinking about what's happening between stations, and also knowing whether a renewal is really happening or not before we get out there. We're hoping to put some additional sensors on the sill mooring. So that has been talked about a lot so far, but there's been an occasional mooring out here just at the bottom, looking at the water that's sneaking into Saanich Inlet, and we would hope to even put a couple of sensors above that into the water column so we could really understand what's entering. And then there's a potential for possible glider missions that would be periodic, looking for gradients in the inlet. Okay, my last slide, I would like to introduce you all to Jeff. He's right here. He's our new postdoc for this experiment. So he'll be coordinating a lot of what's going on here. This experiment only gets more powerful the more measurements we can combine with it. So we are really looking for people who want to work with us, who want to come out and take measurements, and doing intercomparisons of these. So please talk to Jeff and I if you are interested. Thanks.
Any questions for Frank?
Lisa might be able to find it. I don't know somebody else who said the traps, but we had a couple of daycares set up a sequential sediment traps. Took over it 2 weeks, and there was a detail on carbon nitrogen soil export.
I think that would be very
I
exciting.
need somebody to look at the samples, obviously, but something like that would be a nice size trap to handle. Could be hung off the float, and as I say, I think it's 10 sample cups. You could really do some detail on it, maybe monthly or 2 weekly cycle.
Jeff, put Sylvie organizing on your
Yeah, that would be, should be an excellent person to working with Dennis lab to handle some of the samples.
Yeah, I think that would be a wonderful
It sounds like a great program, yeah, something like that.
Mm-hmm. Great.
For Bird in my other life, I do a lot of hunting stuff like that. And working in NOAA, and they have developed a redox sensor. So it's just total redox. Really cute device, a platinum electrode with a silver chloride electrode. And so it just goes up and down at continuous redox. So it's just redox total potential.
Yeah.
And through that, the detections is incredible compared to the old methods. The mapper is a cool little instrument down in PMEL. They loan them out. So you don't even have to buy it. But I can put you in touch with them to get that to you immediately.
Absolutely. And is that something you can like put on your CTV?
Yeah, that's what they do. They just.
Hook it onto the CTV? Yeah.
So
That
I've used it on planting codes actually, to
would, fantastic.
There's lots of people with ideas, so you can do more work.
No, that's. And
It's just a comment with regard to the, what was it? With regard to what we're under. I can't remember if this up, but for years I did profiling. I would use saanich on my way to other places where the profiler actually landed on the bottom, so the sensors ended up about this far away from the bottom. And on that sill, every once in a while, which turned out to find the very, very lowest highs, you would find really dense water, you know, that as you would go like this and then it would go like that. But it could be so close to the bottom, like it could be the bottom foot.
Bottom foot.
You know, so you have to really have a sensor like right on the bottom if you want to identify every single thing. Because that's the densest water.
We have had, I mean, there has been a mooring out there, an autonomous mooring with just bottom sensor, and we have
it
seen.
literally is within that bottom meter. I don't know if it's a foot, but it's very, it's just right at the bottom. So.
I did it closer to that.
Well, the
Yeah.
plan is to keep that one going.
Yeah.
The trick is, I think what Roberta hinted at is very interesting, is that it's not just one water mass that comes in. It's that bottom whole stratified layer that comes in and then finds its density place, comes in along, and it sweeps perhaps even every flood. There's a pulse coming through that, and, you know, the whole
The densest water.
Right, the densest part, right.
Gotta be right bang on the bottom, yeah. Good stuff.
Anyone in the plankton community want to jump in and say what's the sampling during that time frame? I mean, we saw that obviously the euphausids are going through their life cycle. It's mostly the adults in that time. You know, the juveniles from the spring are now coming into their adult life through the winter, and then so that September to June is the mature big zooplankton period for euphausids. But so net toes, I don't know. I mean, I'm just throwing it out there. I don't know what.
Well, yeah, John wants definitely to do
Is John?
that.
That's what John's going to do. Okay.
That's part of it. Okay, look at fatty acids.
Frank.
I'm curious, Roberta, if you've been able to get these measurements on the basin flushing waters that come in for excess nitrogen. Do you have enough data to sort of show what the excess nitrogen flow is?
Not on a seasonal basis. We went out to Harrow Strait once and took a profile there, and we kind of used that as our background value. It was fairly similar to what we see at sort of similar densities offshore, but it is a concern. Now, in the case of Saanich Inlet, the signal is so big that the background, whether the background's 0.7 or 0.5 or 1, is not that big a deal.
It does help to identify where the water is coming from. Is there enough of a signal that suggests that we're seeing California undercurrent water with the
I don't
N-star signal in it, or excess nitrogen?
I don't know. I think that would be challenging to use that as a tracer. To use NAN2 as a tracer with that, but I haven't thought about it that carefully yet.
What we kind of think is we're seeing that as a source water, that it's going to be picking up.
So your plan is, Roberta, you've got effectively the Strickland almost every 2 weeks for that period, and you're filling it up obviously with people and gear and going out for a day at a time, or
A day at a time.
A day at a time. Okay.
That's the idea. Yeah, and we want to do a couple of test cruises this summer to kind of figure out how to do everything efficiently enough and not contaminate all our metals and things like that.
Does iOS, when they go on there, even their, I don't know, Peter Chandler or whatever is doing their Sailor Sea Service, do they do any of the saanich stations on the Vector?
I do not know. I mean, I know Line P usually doesn't dip on the way out.
I mean, I don't know. Yeah, I know Line P usually does one in Saanich, but even if they were to go to like S4,
I don't think anymore, I don't think the program does anymore.
Really? Okay.
Not that I'm sanitary, last time.
Oh, really?
Oh,
I
okay.
don't think Peter Chandler running the Georgia cruise.
We had tried to talk them into, they just leave the dock and do their test profile in saanich to get the bugs
That's
out.
years, yeah.
Okay,
That's the problem.
okay.
And I want to mention too, I mean, I already talked to BBC folks about this, but we'd like to try to marry our time series together to some extent to the amount that we can accommodate taking water for you guys. We want to make that work.
All right.
All right.
Yeah, that's good.
Looks like lunchtime.
Okay, so, lunch again is out in the foyer there, and, the plan is to be back in here. We got, back in here at 1 o'clock, so 35 minutes, and then we'll.
I was just gonna ask you.
I have an iPad at home. I just like, yeah, you did it to yourself. Oh, you did, yeah, but I like you. I don't know, I'm not PC Max. Right or wrong, sorry.
I was just
Yeah, because
Still punches every time.
Yeah.
research. They may have. What's the best side? Some of our core. Yours are the oldest ones. Yeah, I mean, some of them are still going along, but we don't know if the calibration is good. Except for one.
Yeah. Let's worry about they're the ones that keep down, they don't fire power, over the skin, and keep going. But how can we put it back down? It's a stress restriction. So anyway, okay, well, so somebody addressed it again. I agree. We just doing it even when it's. Oh, yeah, that's like, well, especially if it's 30 years. No, it's probably a good thing, I think, because we shall, we shall, the straight trade, we get all these things. But when you get stuff, you get benefits, not for the, so. Yeah. Organization. Yeah.
Stuff like that.
Well, no, it's the compound, because we put the upper rate hustlers. And then it's just. Yeah, that's right, that's secret, that's probably that, those people. The commerce is sort of like, benign,
And that's problem for where we have this, it works better and it doesn't. Then we'll go first.
Yes. That's not editor. Well, based
That was why not actually try. Now we really get an expert. I gotta look at it for the video one time. Once you figure out this technique here, I don't know why you figure it out. It's cool. You kind of like do that big, you hear me do that. Because you figure out that I don't get a $10 beat. No, that's right. Stupid. Yeah. Yeah. We saw a single rainbow, but there was like a thing blocking our view. Either, right? This way, I don't know if I can show up. We've got a reserve seat there. Oh, just wait. We got Damien, David, and then me. Do you want mine now or what do you want? One called physics. Next focus. Should be the first one. I'm now experiencing gravity waves. Actually, I've been experiencing gravity waves all the time. Now they've officially been authorized that waves. I think it's a repulsion. I didn't say it was an attractive force. Not to mention. Why did you? Yeah. Very good. I'll make my way up around the time. Yes, yeah, if I can touch, yeah, you're welcome. I'm gonna do. All right, if people migrate towards their seats, daily vertical migration. All right, Kim.
I've got an idea I'm going to float just before I introduce Damien. We had a workshop for the Barkley Canyon group in October, and an idea that galvanized and the community really bought into, and one we thought, boy, that would be really valuable. It may be a lot of work, but it's probably worth doing, is an idea that we, either as a community, and maybe certainly ONC could look at leading this initiative, would be to build an atlas for Saanich Inlet. And it really would be a sort of a multi-dimensional metadata source where you could go and you could see the layers of history, literally sub-bottom through the water column. You could find out what has been done, where, how it's been analyzed, and it could be electronic. I think the idea was that it's really electronic, and you could search through by your interest, whether it's geology or chemistry or maybe microbial aspects. But it's a structured database that maybe has multiple ways of slicing and dicing the layers, but it's really an atlas, and it would be a starting place. In particular, we can imagine we could all possibly go there and learn things, especially outside of our area of specialties. But one of the strengths for Ocean Networks Canada would be that it would be a place that we could point somebody who's new to the community. And they could come in and they could see what's been done and both the history and what's active right now. So I'm just going to float that as an idea. Think about it as we go through the next few talks. And at the end, we can wrap up and we can get your feedback as to whether that's something that might be a good idea.
Right. I thought on that, a starting point might be to have a library of papers.
Right. And
there's a lot of older papers that aren't accessible.
Yes. Yeah, overview article and a bibliography, and not only just a list of papers, but actually links to them because they're now getting inaccessible. The Saanich Inlet study in the 90s did an amazing job of compiling an exhaustive list of the papers that were available at that time. If you go, it's one of these things that's interesting. You might think they're getting less and less available, but in fact, they're now going back and scanning older and older things. But it would be good that it's not just a list of papers, but they could be linked to them, and you would get them. So that's the type of thing that an atlas could have in it. All right, the next week, yes.
Sorry, just along those same lines, I think you were talking about maybe an inventory of all the data sets that we have.
Right, and that would be part of the atlas.
Who has those, so that when we, especially as grad students, you kind of get an idea, you're like, oh, I wonder if anybody looked at that. People that actually go and talk to someone who's looked at it.
And some of the data sets could be centralized. If people don't mind, they could be centralized and made links available. Or they could be links back to the individual labs if they want to hold on to them. Ultimately, if they're links on an electronic atlas, you don't really care where they reside as long as they're going to have a legacy. As long as the links don't go dead, it doesn't really matter whether the files are UBC or UVic or wherever iOS. As long as they don't die, the atlas could have active links to all of the data. Okay, so the next speaker is Damien Rundle, and Damien can come in. We've only got a couple more, and then we'll wrap it up with a final discussion, and hopefully we'll get out. I don't know if there's coffee at 3 o'clock. It just says adjourned. So,
Yes.
Okay, everyone can hear me kind of? Yeah. Okay, so first off, I'd just like to thank Richard, Akash, Dwight for organizing this and for making it possible to come out this week. I hope it's not too disappointing. I'm not going to actually talk about saanich right now. It's been several years since I've worked in saanich, and I didn't want to rehash things I did at the last symposium. So instead, what I'm going to do is talk about, just in general, or with a specific example, actually, of how we can just use these kind of low oxygen environments as sort of natural laboratories to understand N2O processes in the ocean, or under future sort of oxygen conditions in the ocean. So just first off, just very quickly, a bit of, sorry, where am I standing? Okay. Bit of background. So just first off, simply why are we interested in N2O? Essentially, it's a climate relevant atmospheric gas. It contributes both to atmospheric warming and to ozone destruction. The production pathways in the ocean are predominantly nitrification and denitrification. So nitrification N2O can be produced as a byproduct of ammonium oxidation, or through what we call the nitrifier denitrification pathway. So this is where nitrifying bugs actually reduce nitrite to N2O. In denitrification, it's an intermediate in the reduction of nitrate to dinitrogen. So it's both being both produced and consumed. That said, we do often see net yields of N2O from denitrification. So more N2O being produced than is being consumed. So now just kind of briefly touching on this kind of understanding of the role of oxygen, or the role that oxygen plays in N2O production. So when we get low oxygen conditions, below some sort of threshold value in the ocean, we would expect N2O production to really start increasing quite substantially. And we would also expect to see possible major shifts in the production pathways, or the cycling pathways of N2O. So this raises a bit of cause for concern because, as we're aware, oxygen concentrations in many regions of the oceans are decreasing. So it's very likely that at some point through this century, we're going to see N2O production really start to possibly ramp up quite largely. Okay, so we've got this understanding that, okay, decreasing oxygen concentrations will eventually lead to an increase in marine N2O production. Most of our understanding of this process has come from laboratory studies, and these studies really don't mimic what, the kind of conditions we see in the field. So I think, the point to make at the moment is that really that oxygen threshold, which induces a substantial increase in N2O production, causes major shifts in the predominant production pathways, is really poorly constrained, especially when we talk about these kind of natural mixed assemblage conditions in the field. So I think the point is that we really need to take up questions, these types of questions pertaining to N2O processes. We need to take them out of the lab, and we need to start addressing them in the field under natural mixed assemblage conditions. So to do this, what we really need to do then is locate these marine regions which are characterized by naturally occurring extreme DO, or dissolved oxygen gradients. Put another way, we just need to locate regions that have these very, very low oxygen conditions where we might start to see major shifts in N2O production and the N2O production processes. And of course, one of these is saanich inlet. Now, over the last few years, I've been working in a number of regions looking at this, so the Eastern Tropical Pacific, the Eastern Tropical Atlantic, the Baltic Sea, and then also we've been doing quite a bit of work in low oxygen eddies, which are forming in the Pacific and the Atlantic. And the types of measurements we're looking at to try and address some of the questions pertaining to N2O processes include bulk N2O concentrations, N2O isotope signatures, been developing and employing 15N tracer techniques to measure N2O production via the different pathways. And just as a side note, Krista Marandino and I have also recently been developing the eddy covariance technique to directly measure N2O fluxes for the first time. We were just able to successfully deploy this technique on a cruise we had in October of last year. So that's going to really help us sort of understand if we're going to see a lot more N2O being produced, how much is going to be fluxed out to the atmosphere. So the area I'm just going to touch on today is a low oxygen eddy from the Eastern Tropical North Atlantic, because I think it's just a pretty simple, straightforward story to kind of tell and give you some examples of how these low oxygen systems can really be a valuable tool to understand not just N2O processes, but biogeochemical processes in general. Okay, so give you a bit of background about one of these eddies. So here we've got the Cape Verde Islands off the west coast of North Africa, and the X marks the location of the Cape Verde Ocean Observatory, or the CVOO time series station. So back from about 2006 to 2013, some colleagues of mine at Guiomar, led by Johannes Carstensen, had a mooring program out at the CVOO time series station, and one of the instruments they had was an optode at about 40 meters depth. And when they started looking at these results, what they found was that every so often in 2007, 2010, and 12, there were these extreme decreases in oxygen concentrations, right up in the near surface waters at about 40 meters depth. Now, what they realized when they went through the satellite imagery data was that these periods of low oxygen events occurred at the same time as eddies were propagating through the CBO time series region. And you can just kind of see this up here. So we've got these eddies forming off the Mauritanian upwelling system, and then they're propagating westwards past the Cape Verde Islands. So the realization was then that some of these eddies really must be characterized by much lower oxygen concentrations than the surrounding waters. Because also remember, we're dealing with the Atlantic Ocean here, which is a pretty oxygenated ocean. So when I first got to GMR 2013, or early 2013, it was thought it'd be pretty cool, multidisciplinary group together, and go out and do a preliminary study of one of these, sorry, I'm walking all over the place, do a preliminary study of one of these eddies. So in late 2013, we started to use satellite imagery to start identifying a potential low oxygen eddy candidate. We then followed that up by conducting glider surveys in early 2014 of this eddy candidate that we identified. And here you can see some of the results from that. So we've got salinity, temperature, oxygen, and nitrate here. What I really want to focus on here is the oxygen. So you can see that in the core of that eddy, there was this ball of just super low oxygen water. So these results really confirmed to us then that yes, we were dealing with a low oxygen eddy. So following that, a couple of us went down to Cape Verde in March 2014 with the goal of going out and conducting some shipboard sampling of this eddy. That was easier said than done. We spent about 4 or 5 weeks down there trying to get out. The ship was in pretty rough condition. Multiple times we turned around and come back, and compasses were breaking, engines were breaking. We finally got out and were able to spend a couple of days in one of these eddies and get some sampling done. So I'm just going to focus on the N2O portion here. So take a look here at this figure. So I've just plotted oxygen concentrations and N2O concentrations at 2 stations that were inside the sort of core of the eddy in red, and 1 station outside of the eddy in black. So the oxygen concentrations outside of the eddy were very typical for that region, the kind of concentrations we would expect to see. When we got inside the eddy, however, you can see that in the upper 100 meters, the oxygen concentrations just dropped right off. We were getting down to just below 5 micromoles of O2 per liter. So this is a massive decrease in oxygen concentrations. When we look at the N2O data, what we see is that outside of the eddy in black here, the highest concentrations we measured were about 35 nanomoles of N2O per liter. This is, again, what we would expect from already having done a lot of work out of the Cape Verde Ocean Observatory. And this is also within the range of the kind of highest concentrations we see anywhere in the Atlantic. Inside that eddy, however, the N2O concentrations ramped right up. We were getting up to almost 120 nanomoles of N2O per liter. So this is by far the highest N2O concentrations ever measured in the Atlantic Ocean. And I think what I really pointed out was, yes, okay, we've got this system that's really low oxygen. The N2O production appears to have really ramped right up. So yeah, we can now maybe use this as a model system to start tackling the question of what is that oxygen threshold where we see an increase in N2O production. So unfortunately, I had hoped to do 15 tracer experiments on this cruise. It ended up really not possible with the conditions on the boat. So to tackle this question, I plotted this figure here. So we've got del N2O on the y-axis, and AOU, or apparent oxygen utilization, on the x-axis. So delta N2O is essentially an estimate of how much N2O has been produced within a parcel of water since it was last in contact with the atmosphere. And AOU is an estimate of how much oxygen has been consumed within that same parcel of water. So we kind of look at this as depicting N2O production versus oxygen consumption. And when we plot this data, we see that we've got 2 pretty distinct relationships. We've got this first relationship here, which is occurring at oxygen concentrations above about 20 to 22 micromoles of O2 per liter. And you can see that the N2O production is low versus oxygen consumption. When we get dropped below 20 micromoles of O2 per liter, the N2O production versus oxygen consumption just ramps right up. I mean, this is a massive increase. So, this is really kind of giving us some insight into that oxygen threshold, which causes that increase in N2O production. So we can take this one step further, the interpretation. If we look at the slope of these relationships, we can say that above about 20 micromoles of O2 per liter, we're seeing about 8,500 moles of oxygen being consumed for every mole of N2O that's being produced. When we drop below 20 micromoles of O2 per liter, we're all of a sudden seeing only 170 moles of oxygen being consumed for every mole of N2O that's produced. Put that another way, and that's about a 50-fold increase in the amount of N2O produced versus the amount of oxygen being consumed. So I think these results are really cool. I mean, this is a preliminary study. This is, a limited data set at the moment. And I do have plans, a 3-year funded project to go back and investigate these eddies in more detail. But I think this really gives us that sort of first insight into the potential future N2O conditions in the Atlantic Ocean if oxygen concentrations continue to decrease in this region. So now we've been able to identify that threshold, which induces a substantial increase in N2O production. And now we've also been able to get some insight into the amount by which N2O production may increase. So the other thing I wanted to look at was, also how is the oxygen maybe, or how the conditions inside the eddy impacting the production pathways of N2O. So to do that, I was looking at the isotope signatures, and here I've just plotted data from inside the core of the eddy and data from outside of the eddy, just to try and highlight what's going on. So we've got the del 15N of N2O, we've got the del 18O, and the 15N site preference here. If we just block out that 100-meter mark for now, where we're seeing the lowest oxygen concentrations, and focus on the depths above and below inside the eddy and compare it to outside of the eddy, one of the things we see here is that there's a consistent sort of, fairly consistently, the del 15N and the 15N site preference is lower inside the eddy than it is outside the eddy. So these isotope signatures are telling us that in both regions, nitrification is the predominant production source. But inside the eddy, we tend to have a bit more nitrifier denitrification producing N2O. So, and this is very likely due to those sort of lower oxygen concentrations. When we get into the core of the eddy, however, or the core of the oxygen minimum zone, at 100 meters depth, where we were getting, so this data point is kind of representing a transition zone between about 8 and 4 micromoles of O2 per liter. And what we're seeing is we've got one of the lowest del 15N values of N2O that have ever been measured in the ocean. We've got a moderate increase in the del 18O, and we've got a fairly substantial increase in the 15N site preference. This is indicating to us that we've had now this major shift to where, especially the del 15N value here, is indicating we've got this major shift from nitrification pathways being the predominant production source to now denitrification is producing N2O. And the increase in the 15N site preference suggests that we've also got partial consumption of that N2O by denitrification as well. But the production source is definitely outlaying the consumption source. So again, I think this is really cool data, because this is really now giving us insight into the oxygen threshold that all of a sudden turns on denitrification. And shifting from nitrification being the predominant sort of cycling pathway for N2O to denitrification starting to cycle N2O. And just to sort of highlight this on this figure here, so here we've got AMOA gene abundance and expression, and the same for NERS. So AMOA is a gene involved in the nitrification pathway, and NERS is the gene involved in nitrous oxide production through the denitrification pathway. So if we just look at the expression, looking at the triangles here, symbols here, we can see again that 100-meter depth mark, where we get those lowest oxygen concentrations, the expression of the AMOA gene is just dropping right off, whereas the expression of the NERS gene is really just ramping right up. Again, providing additional support to this conclusion that denitrification all of a sudden became a very important cycling pathway for N2O. Okay, so just to sort of summarize, so again, I think these types of low oxygen eddies or low oxygen natural laboratories, just like saanich inlet, are ideal locations to really go out and investigate these kind of how N2O processes will respond to future oxygen conditions in the ocean. We can really, and it's not just N2O. I mean, we can go out and use these kind of environments, saanich inlet, these eddies, the Eastern Tropical South Pacific, all these regions as these areas to go out and understand how biogeochemical processes in general will respond to future oxygen conditions in the ocean. So just to sort of summarize, in this eddy study, we were able to start identifying that tipping point where N2O production really starts to ramp up. and we were able to identify that as being around about 20 micromoles of O2 per liter. We're also able to identify the potential degree by which that N2O production may increase. So we were seeing 50-fold increase in the amount of N2O being produced versus the amount of oxygen being consumed. And then we were also able to start identifying that threshold where we see a major shift in the predominant cycling pathways of N2O. So somewhere between that 8 and 4 micromole of O2 per liter range, we saw denitrification all of a sudden becoming a very important pathway for N2O cycling. Now, just as a side note, although we were seeing that evidence for denitrification, it certainly did not appear to be important for N-loss processes at that point. So it was important for N2O, which is we're dealing with on the nanomolar scale. It did not appear to be important for fixed N-loss, which is, you know, we look at sort of on a more micromolar scale. So I think it's just important to really highlight, yeah, these systems really are these great places to go out and study. And I think it really kind of heeds Luke Cottespotty's core science from 2010 that future experiments really need to go out and focus on the controls for marine N2O production and focus on these N2O production hotspots where there's a lot of activity going on and we can really learn a lot about these processes. Okay, thanks.
Questions? Yes, Frank.
Right.
Do you wonder if the oxygen threshold for denitrification might also be correlated to the amount of nitrate available? I think microbes can use oxygen and nitrate and get just about the same amount of energy from them. So I'm wondering if the oxygen threshold is very much tied into the amount of nitrate available. In other words, if there's very little nitrate,
yeah,
you're going to take oxygen down much lower until you start using nitrate.
If there's very little nitrate.
That's right. If there's a lot of nitrate, you might see a higher threshold.
I don't think so. Just if I compare it to, so you're saying if there's more nitrate, we'll see a lower threshold for oxygen.
No.
Go the other way around. Okay, then no.
What I'm going to imagine is about 20 of each.
Yeah.
Microbes might use either one. You have 20 micromoles of either. This
Yeah.
50 and 10, and they're going to choose the 50 source.
Yeah, no, so here we had really super high nitrate concentrations. I mean, really quite high inside that eddy. When I compare it to other regions where we've got lower nitrate, it doesn't really, at least from the N2O perspective, it doesn't seem to be changing a lot, the story. I mean, we're seeing fairly similar O2 thresholds. They do vary, but I think they're varying more because of the community composition. You know, in an open ocean environment compared to maybe a coastal environment, or, you know, an upwelling environment of the ETSP region. But maybe, but I haven't really looked into it yet.
So based on your knowledge of saanich inlet, are we looking at, for these sort of transitions or sensitivities, do we need to look at either spatial or temporal boundaries? Are there horizons that you really want to go and say, you know, that's the place we should do higher sampling because either spatially or temporally?
Yeah, you don't look at your option profiles to start with. Okay. And I think you really want to be focusing, you know, based on results from other research, I think you really want to be focusing on that somewhere between about 25, 30 micromole of O2 per liter and doing very fine resolution sampling below that. I think it's really important that we don't just limit this work to one region, because there are going to be other factors which do influence this. And, you know, I don't think those will really bear out until we have done this kind of work in a number of different regions. In saanich, I think, you know, it's very important, and I think, you know, David will show some of the N2O data from saanich inlet next. You know, in the open ocean, we get these really tight relationships between N2O and AOU. I mean, we see it in a number of different regions. When you get into coastal environments, it becomes a lot more messy, I think, because you're getting, you know, a lot of N2O coming out of sediments as well. So I think it's really important to, at some point, hopefully, you know, get 15-inch tracer techniques going in saanich inlet so you can directly measure the rate of production. And, you know, as you go down through this sort of low oxygen boundaries, and you can then really just nail down that sort of oxygen threshold. It's really kicking everything into gear.
Just wondering if you looked at all at microbial community structure or if you have plans to for your next.
No, we haven't in detail, and it's certainly something I would like to find anyone that's maybe interested in joining along and doing some of this work. I don't have the money to necessarily fund a whole bunch of work, but if anyone else is interested in coming along.
Sequence tags or something, you're not looking at, you're already collecting your DNA for your PCR. So
Yeah.
It's about the same amount of DNA, you don't need a lot.
Yeah.
That's gonna be really interesting if we look at different places. Is it the same groups? Is it different groups? Is it different OTU, different species of the same group? That
Exactly.
is going to be our different species within the same group behaving the same or differently under similar conditions.
Yeah, no, I think this is key. It's really key to also understand then how different systems vary, because what we see in the Baltic Sea is not necessarily what we see in another coastal environment. What we see here is not exactly what we see in the Eastern Tropical South Pacific. And I think this is all being driven by different communities. No, that's why I say it really didn't look like it was becoming important yet for fixed end loss on the micromolar scale. We looked at the NSTAR, we also had N2 access or biogenic N2 measurements, and you really didn't see anything there. I think if we drop down a couple more micromolar or two per liter, then we all of a sudden see that kicking into gear as well. So.
All right, thank you very much.
Thank you.
All right, our next speaker, David Capel, 8-year time series of N2O research. You hear me? No? Let's talk like this. That's right. Okay. Sure. Cheers.
Okay, hello everybody. I'm David Capel. So I just want to take a second to thank you for inviting me to this talk, and thank you all for coming. At the last saanich symposium 4 years ago, I was just starting my PhD with Philippe Tortel, and I really didn't know a lot about saanich inlet or anything. I was really interested in studying greenhouse gases in the oceans because I felt like the ocean is a big mystery in terms of greenhouse gas cycling. And so Phil, he showed me this cold room we had back at UBC, and it was full of about 1,000 samples. And it was going to be my job to analyze these and then make sense of them. I'm sure some of us have probably been there before. So I was very optimistic at the time, and now, I thought it would be really great. I'd come up with these great things. And 4 years later, I think I have more questions than answers, but I guess that's how it goes. I want to thank Damien for his great talk because he basically is going to allow me to skip through the first 3 or 4 slides of my presentation. He already talked about how nitrous oxide is kind of a byproduct during nitrification, and you can have these sort of short circuits between nitrification and denitrification, which can also lead to the production of N2O, and that denitrification is a source of N2O at times. At other times, maybe it's a sink. So he talked about the importance of understanding oxygen thresholds where N2O production becomes enhanced. And what we know so far is that, as Damien said, in low oxygen waters, that's where you tend to see the most rapid N2O production. So, of course, when you're talking about the expansion of oxygen minimum zones, people have been talking for a number of years already about how that's going to maybe lead to the enhancement of sea air flux of N2O and production rates. And of course, oceans are an important source of nitrous oxide already. They supply between 1/4 and 1/3, based on recent estimates, of the total N2O to the atmosphere. And nitrous oxide is one of the most important greenhouse gases after carbon dioxide. So yeah, I've already talked about some of these. And then, of course, saanich inlet is a great place to study how changing oxygen availability will affect N2O cycling in a natural environment. And that's what we're trying to do. We're collaborating with the Helm Lab. We're doing a great work with metagenomic data and microbial data. And we're trying to combine some physical measurements of N2O in order to characterize how the entire ecosystem is responding to these changes in oxygen availability. And so we're hoping that we can sort of make some conclusions and maybe extend them out to sort of a global ocean model to get a better handle on how oxygen availability is going to change N2O cycle. So as part of the Hallam's time series and my advisor Philippe Tortel's work, this time series was established back in 2006. I think we started taking N2O samples and methane samples in 2007. And so yeah, I just want to take a second to thank everybody in the Hallam Lab for all their hard work over the years to help get these samples. Couldn't have done any of this without their tireless efforts. So I'm really grateful for that. So my main research questions are, because we have this time series, this wonderful monthly time series, we can look at things that maybe don't get captured by more discrete summertime sampling things. I think Roberta was talking about that, how a lot of the work that's been done in Saanich Inlet is done during the summer. And so we don't really know what's happening in the winter. So that's important when we're trying to describe total oceanic changes and generate annual budgets. And then of course, as I talked about already, we're looking at the effect of changing oxygen availability on nitrous oxide cycling. And because we're starting to get into almost 10 years of time series measurements, we can start to look at long-term changes in N2O cycling and fluxes in Saanich Inlet. And then, as we'll see towards the end of my talk, I'm actually going to try to extend our observations over the last 8 years to some of the previous work of measurements of oxygen and nitrate in Saanich, and try to maybe see if there's something there that's been going on for longer. So this is a snapshot of our data. We've all seen plots like this before where you have depth on the y-axis, concentration is indicated by color, and then time across the x-axis. And here in these 4 plots, I'm showing oxygen, nitrate, nitrous oxide, and hydrogen sulfide. These are some of the key parameters, of course, that are indicating the level of oxygen in the water, and nitrate is a good indicator of nitrification happening and denitrification. And we look at the N2O data, you can clearly see that it closely tracks, at least in the deep basin, the annual cycle of renewal and then deoxygenation. There's definitely some interannual variability, and so my first sort of step was just to cut through that noise and look at a mean annual cycle. So we basically condensed that time series into a single year to get an idea of what the mean annual variability is. And so again, the oxygen cycle is very clear. You have anoxia developing in the deep basin during the summer, and then that allows denitrification to occur in the deep basin, which consumes nitrate and nitrous oxide. And then during the fall renewal period, typically in September to October, you have the reoxygenation of the deep basin, and that renewal water also brings in nitrate and nitrous oxide. And so one of the questions we had that we're trying to answer with this time series also is, does that renewal actually stimulate N2O production? Does the resupply of a little bit of oxygen actually inhibit denitrification perhaps, or complete denitrification, and sort of lead to a very strong pulse of N2O? And actually, I'm not showing that data here, but I'll basically say it doesn't look like that's happening. It looks like following renewal, N2O is not produced in the deep basin, it's just consumed. So one of the other interesting things we see in the mean annual cycle, which has been talked about a little bit already, is this seasonal shoaling and deepening of the oxycline. And that's shown by the deepening and shoaling of the 90 micromolar oxygen contour here. And that seems to, if you look here, I've taken the oxygen and nitrous oxide contours, and you can see that that shoaling of the oxycline corresponds to a period where we have very high N2O concentrations near the surface. And we also calculated N2O fluxes over the course of this 8-year time series and did it on a mean monthly basis. And you can see that this time of year, the sort of post-renewal period, also corresponds to the periods where you have very strong N2O fluxes. So this shoaling of that hypoxic water both appears to support high N2O production and flux. And that's an important finding. I think it's something that is being predicted by models, and the expansion of oxygen minimum zones is supposed to lead to this, and here we have it happening in Saanich Inlet. Now, it's difficult to say whether that flux is also maybe driven by upward transport. Roberta talked about how renewal can push that anoxic water upwards. It may be pushing nitrous oxide upwards as well, but it's hard to sort of distinguish between the two. So another question we had, of course, is just trying to distinguish between N2O production from nitrification and partial denitrification and other sources. And so what you expect to see, and Damien kind of showed something similar to this, what's often done when you're studying N2Os, you plot it relative to apparent oxygen utilization. And this is kind of the trend you expect to see, where as oxygen is consumed and you move left to right, the amount of N2O in the water column increases. And that the slope of this curve also increases over time because you have additional yields of N2O under low oxygen waters. And you also have the contributions of partial denitrification, perhaps, when oxygen concentrations are very low. Now below maybe, again, this threshold isn't clearly defined, but below somewhere around 5 to 20 micromolar, N2O is in fact consumed by denitrification, so you expect to see it drop. So if this is what we expect to see, now let's look at what we actually see over the entire 8-year time series. A lot of scatter. Damien kind of alluded to this already. We spoke about this briefly. When you compare this to an open ocean region where they've done these types of plots, Saanich Inlet, there's a lot of variability over this line. The yellow line indicates the linear regression of these points between 0 and 270, kind of just trying to exclude those points where N2O is consumed by denitrification. So we really don't seem to see that inflection in the curve under very low oxygen concentrations like you would expect if you were getting partial denitrification or higher N2O yields. And so this is kind of puzzling, but again, there's a lot of noise, so we have to try to cut through the noise. And one of the reasons we think that there is so much scatter in this data is that there's actually seasonal variability in Saanich Inlet in the relationship between N2O and oxygen. So this plot on the left shows the N2O versus oxygen concentrations for all our data collected in March in black triangles. And in the open circles, we have the same data, but for only samples collected in October. And what it implies is that you get higher N2O production rates and you see higher N2O concentrations at a given oxygen concentration during the fall than in the late winter, early spring. And so we calculated the mean slope for each month of this relationship and plotted it over time for the entire 8-year time series. And you can really see this gradual transition from gentle slopes to very steep negative slopes over the course of the spring and summer. The causes of this are kind of unclear. I think it basically implies that you have higher yields of N2O during the summer than in the winter. Maybe it's a sedimentary signal. Maybe it's due to a higher abundance of organisms in the water column, microanoxic zones. Frank's got a question. Go ahead.
Maybe at this point it's good time to ask the question. Do you have ammonium data?
We do.
Yeah, because ammonium does accumulate below the zone. In summer, right? It sort of progressively accumulates.
Well, it accumulates in the deep basin, definitely in the summer.
I'm thinking just below the zone. You can't use it. You got to turn over nitrogen.
You have some accumulation of ammonium, yeah, just below the euphotic zone a little bit, and then it gets consumed about 10, 20, 30 meters below that. So you have this sort of...
Yeah, but and turned over, so it's oxidized. So the oxygen zone.
Yes.
I'm wondering if that's the source of increased production.
Well, that might be.
Summer and late summer would be the oxidation of ammonium.
Right. I haven't looked at seasonal differences in that ammonium peak below the euphotic zone. So that's an interesting point. I'll have to look into that. Thank you. Now, as I mentioned, we have a lot of scatter in saanich inlet, and another interesting fact about this, the relationship between N2O and oxygen, is that it's a much gentler slope than you see up just a few kilometers away off the west coast of Vancouver Island. So I've also collected, as part of my research, a bunch of samples on the La Perouse cruise on the shelf of Vancouver Island, and you can see that those data tend to make a much steeper slope. Whereas saanich inlet is very gentle. And also, I guess, off the coast of Vancouver Island, you don't see that drawdown of nitrous oxides, indicating that there's not complete denitrification happening there. So another thing that Damien talked about that I also am looking at in saanich inlet is that threshold, that critical threshold of oxygen, where you see a shift from N2O consumption to net N2O production. And so what I've done here, I've looked at that problem in two different ways. In the panel on the left, I've calculated the mean N2O concentration in each 5 micromolar oxygen bin between 0 and 300. And you can see that we get the most N2O typically between about 20 and 30 micromolar. So below that, that's where you start to see N2O consumption by denitrification. And then another way we've done this is we've just plotted kind of a P versus I curve to our raw data. And that gives us a slightly different result, but really the most of the N2O consumption seems to happen below about 10 micromolar oxygen. And the reason we don't have values below 3 is that's about the detection limit of our oxygen sensor. Okay, so shifting now to some intra-annual variability and long-term trends. This is the time series of the mean water column concentrations of oxygen, nitrate, nitrous oxide over our time series. We've also plotted N2O flux over time in the bottom panel. So what we're seeing, I think, is some evidence that definitely we see a decline in oxygen over time. We've talked about that being observed by others in saanich inlet. But that seems to be related to an increase in nitrous oxide in the upper water column, as you would expect. And that also, importantly, is being translated into increasing N2O fluxes from saanich inlet. Now, this is only an 8-year time series, and the only other nitrous oxide measurements available from saanich inlet were done in 1978 by Yuval Cohen. So we can't really compare this to a longer term, more meaningful time series. But because of the clear relationship between N2O and oxygen, we can look at long-term trends in oxygen over time and get an idea of maybe that because we see long-term declines in oxygen dating back to the 50s in saanich inlet, maybe we could expect that nitrous oxide has been increasing since that time as well. And that's an important finding, I think, that can't be understated. We also see changes in salinity and temperature over time in saanich inlet, maybe indicating warming, increased stratification. Another possibility might be that we're seeing changes in the relative contributions of the California undercurrent to saanich inlet, which we know that there's a huge N2O hotspot off the coast of California, and that N2O has been shown to be transported over long distances, and that might be having an effect of saanich inlet, but it's really, this data isn't really adequate to actually answer that question. So to summarize, we have a really great time series from a great model ecosystem, and we've been able to answer some of the questions. I think we have a lot more questions to answer, though, that will require a lot of future work. We seem to sort of conclude that most of the N2O in saanich is probably from nitrification. There doesn't seem to be a spike in that N2O yield under low oxygen concentrations. So maybe not a huge signal from partial denitrification or nitrifier denitrification. Definitely stronger production and flux during the summer. And the seasonal variability in that N2O to O2 relationship is important. A lot of people are trying to predict N2O concentrations just by measuring O2 and relying on that clean linear relationship between N2O and O2. And I don't think that's going to work in saanich unless you really look at it on a seasonal basis. And then finally, yeah, we're seeing some evidence maybe of long-term trends that are sort of confirming some of the predictions of models and N2O researchers. So course, this time series is really just based on physical measurements of absolute concentrations, so without actual rate measurements or measurements of inputs from sediments or microanoxic zones. It's difficult to really determine some of the rates and the relative sources of different processes, but I think those would be useful things to do in the future. And of course, because we're collaborating with the Helen Lab, and they have a great library that spans this entire time series of metagenomic data, we're going to be looking at the distributions of known N2O cycling organisms and genes in the water column and how that relates to N2O. So, yeah, and then the final thing is that we also have methane data. We have a beautiful time series of methane in Saanich Inlet, and there's some really interesting stories there. So if any of you are interested in methane, I could give another whole talk on that. So thank you very much to everybody who helped make this happen, and thank you all.
Any questions for David? Amy?
Yeah, good talk. Just a comment and a question. The indirect process, the sandwich particularly, maybe quite underestimated simply because it's very productive and you've got a lot of surfactants on the surface which are going to dampen that chain. And I think it'd be very cool if you could do direct flux measurements, but I mean, unfortunately, it gets harder to this round than the ocean to do those. But we're trying to come up right now with some better ways maybe to define K based on some of the other environmental biochemical signatures. Maybe that would be an interesting comparison. Additional estimates and the revised K.
I agree. I think, can I just respond to that before we go? So the flux measurements were really kind of a sticky thing, and I think we're going to run into some problems with that because most of our flux measurements are based on measurements taken at 10 meters, concentration measurements taken at 10 meters. And as we know, the mixed layer in Saanich Inlet is maybe 3 meters, right? So we do have some samples from the mixed layer, and from about 10 or 11 months. So we calculated a sort of mean difference between 10 meters and the mixed layer concentration to estimate what the N2O concentration is in the mixed layer. Again, surfactants we haven't considered, but I think the important thing to take away is that the increase in flux over time, as opposed to the absolute flux values. Yeah.
Then the differences between like March and October, for example, and the N2O to outro relationship. I mean, working with Steve's group as well, is there any big shift in the community? Because I guess the different times you can have an oceanic community being introduced into the inlet versus maybe more coastal. I'm just wondering if the actual overall community is changing or that's making
Maybe Elise or Monica could speak to that. I mean, we're going to be working together over the next few months looking at those types of things, but
Yeah, I think we've been looking we've done that with the methane, but we haven't done it with the nitrous oxide.
it's something we're going to look at before we publish this data, for sure. One of the other interesting things that's important about that is the difference in the average oxygen concentration in that upper 120 meters is much lower during the summer than in March, right? So that might be contributing to it, and so you might have high N2O yields due to low oxygen concentration, and that N2O might just be diffusing or being mixed throughout the water column, which would maybe contribute to that.
All right. Thank you, David.
Thank you.
No, I did not do the agenda, so the fact that I'm up last wasn't my own doing. That's okay. I'm going to, actually, this could well have been something we did earlier, but I've actually learned a little bit here and there, and there's some emphases I'm going to put in here for me and some of the talks we heard today. So this is a little bit of a wrap up to say, well, we've heard about some of these processes, the deep water renewal, it shows up in everything. Roberta talked about the intermediate role, Coriolis pulling it up to the right side of the inlet, perhaps some recirculation. And I'm just going to pull some of those things together as maybe a final framework as we go into the final discussion to look at the experiments coming up and what we're doing. So this, we've all been seeing this quite, this chart. Here we are in Saanich Inlet and the characteristics. It's a protected fjord. I think Steve mentioned that earlier. If there are winds, and they're not the dominant winds, they're not the thermal winds, it does get protected from the big winter storms from the south. But if there are any winds in Saanich Inlet, they are totally north and south. They're on the axis of the inlet, obviously, but they're rare. The sill depth around 75 meters, and the maximum depth considered the sill is here, roughly, but it's shallow all the way through Satellite Channel. So it's a broad sill, but the edge of the sill is right about here, starts to deepen up right at the head of the inlet. The bow wave comes down, and the deepest point of the inlet is just off here, Swallow Reach. There's no significant estuary circulation in the sense that Gold Stream down here is almost non-consequential as far as circulation is concerned. As pointed out, some of the other rivers, certainly the Cowichan and the Fraser, are more dominant, and as a result, it actually gets fresh water from outside coming in at the surface, and I'll just talk about that in a moment. The tidal currents in Saanich Inlet itself are not that important in the sense, or at least they're not that strong. They're important at the mouth because every flood, the water's coming in and every ebb, the water's going out. So when we do get a renewal event, it's going to be coming in predominantly on the flood tides. So they're important then, but the most important aspect of the tides regionally are the fact that it is the strong tidal currents out in, we'll call it Harrow Strait, but among the Gulf Islands that is really providing a lot of the mixing. And I'm going to talk a little bit about that shortly. So the currents in the main part of the basin are relatively weak. There is generally, in some of these basins, a rather weak anticyclonic cyclonic circulation, anti-clockwise circulation. So things do tend to come in, and they'll have the right side, and they'll come around, and we bring them around. I don't have any of the plots. Jody and I were involved with a dye release experiment a few years ago, and we put some dye in here. Again, imagining it at sort of the mid depth. If it were to catch some of that denser water coming in, where would it go? And we had the vertical profiler, the vertical toad body, or the toad body traveling up and down here to try to capture it. And Jody was out on the Tully with his vertical profiler and covered much more territory. And we felt that when we were on the Strickland here with the toad the toad body at fixed depth, we had trouble sort of capturing the dye, and we thought it was very weakly dispersing. But then Jody brought data off the Tully, and he showed that in fact the dye had stirred. And so I'm going to just point out the difference between mixing and stirring, in the sense that we often think you can actually imagine that mixing is three-dimensional, but it does involve very dramatically the vertical component. And so when you have mixing, you're really talking about diapicnal transfer across the stratification, the background stratification. So you're mixing things that are up in the water column down, and things that are down in the water column up. So that's a distinction of mixing, whereas stirring might be imagined as more lateral. So it comes in on a density surface at one depth, and it stirs and mixes laterally. So you can, if we use those terms or think that way, that really can help you understand some of the lateral versus vertical processes. So mixing, more vertical; stirring, more lateral. And so the mixing is primarily happening out in the Harrow Strait or the Satellite Channel, and the stirring is happening in the inlet. So vertical mixing weak, some internal wave activity, and mixing more lateral stirring. Density intrusions, these are the deep water renewals we talk about, and we've seen these all in all of these signals for the time series. You have these annual bumps in all of these time series, and they're dominated by the deep water renewal, and I'm just going to talk about that in the next little slide here. So here's sort of the two sections we have. We have the chart showing the stations. What is this? S1, 2, 3, 4, 5, and 6. Here's roughly, I didn't want to put it right on top, but our buoy profiler station on the observatory is just south of station 4. The node is up here, just out in Pat Bay. and we have had an autonomous mooring on the bottom up near the entrance at the sill there. It's probably just a little east of S6 here. In the profile sense, and this is going all the way out into Harrow Strait, so all the way out here till the deep water of Harrow Strait. Coming over, here's this satellite channel. This is this broad, shallow sill at about 75 meters depth. And then we come into the Pat Bay area, down to the buoy profiler here. The deepest part of the inlet, again, is about here. And then we have this narrow tongue of the inlet. And that's all this part here, it's relatively narrow. So you can almost imagine that the inlet in a pinched off way at Squally Reach is sort of getting pinched off here. And there really isn't much volume of water in this part, even though in this transect it looks like, oh, there's a lot of water here. This is the narrowest part of the inlet. So this portion here is the main part of the basin. And we did try to locate the buoy far enough in that it wasn't seeing absolute chaos at the front end of the inlet, but represented the basin of the inlet. And I think that hopefully in time that will be borne out. So I'm going to now just talk a little about using this diagram at the bottom here to talk about some of this circulation processes. So in my view of the world, a lot of the dominant changes, whether they be major changes in salinity, the oxygen, or fresh water, they're really large-scale external processes to the inlet, and the mixing that regulates that to some extent is also external. So I'm thinking we have sources for fresh water that's primarily rivers and rainfall outside of the inlet, the Cowichan River, the Fraser River, for example. Or if we're bringing in dense water, it's stratified by salt, and really the source for the salt water is outbound. It's off the west coast of Vancouver Island, coming all the way in through Juanifica Strait and getting mixed up in Harrow Strait. I've cartooned on this section the mixing outside of the inlet. This is really mixing already starting, if you like Steve pointed out, at the sill that's south of Victoria. It comes into Juanifica Strait. The first part of the mixing happens south of Victoria, and it goes into Harrow Strait. Very strong, dense topographic features in Harrow Strait. We get top to bottom mixing, and then we get into the Gulf Islands and Satellite Channel. So these sort of vertical mixing processes are really representative throughout the entire Gulf Islands. And Roberta was absolutely right. I tend to also use, having looked at this data, if we're looking for a proxy for mixing, use the currents in active paths as a really good proxy. There is some sort of lag, if you like, in the currents versus the tidal height. But looking at tidal height gets very confusing as to trying to distinguish the spring and the neap cycles. Look at the strength of the currents in active paths, and you can even, if you want to, sort of take a daily average of the currents in active paths, and you'll get a good picture of the fortnightly cycle. The tidal currents are really what's driving the mixing, and you can get strange anomalies in the height, which may not vary tide strongly to the mixing, but the currents are what's driving the mixing. The other thing about the tidal mixing is, I mentioned it earlier, we actually get in 1 month sort of 4 spring neap cycles. And they go through an interesting transition. You have a very strong spring once a month. You then have a neap. The following neap after the very strong spring is the stronger of the neaps. You then have a weak spring, which is just a bit stronger than the previous neap. And then you have one very weak neap tide. And then it goes back into a very strong spring tide. And it's that one very weak neap tide which is going to give us predominantly the deepest renewal events. So I'm going to talk about that right now. So what we end up having is the neap tides, we actually have the weakest mixing, and we actually have 1 weak neap tide a month that is going to have the minimal amount of mixing. And that's when we get the very deep water coming in and replenishing. It sinks right to the bottom and it pushes whatever's at the bottom, it displaces upwards. I've drawn, here's a cartoon. Not only is it roughly the oxycline, but it's certainly also where Venus has its instrument platform at that 100-meter isobath. So we see that dense water flush by. It doesn't necessarily mean that that's the water that resides there. When we have a deep water renewal, it passes by and goes into the deep part of the inlet. We also have renewal events that are coming in intermediate depths at various other levels of mixing. So sure enough, we have once a month very weak mixing. At other stages of the tide, the mixing is a little bit stronger. And every time it's a little bit stronger, the water that comes into the inlet is perhaps a little less dense. That's certainly true in the summer when we have the salt coming in the upland. So gradually we go up through the other neap. For example, we might have intermediate here. And then when we have the spring tides, the stronger mixing is when we might get some renewals even at the surface or at mid-depth. Surface water that's coming in needs to be fresh. By definition, it's the lightest water. Salinity is the stratification is dominated by salinity. So when there is a lack of mixing, that's when the water at the surface is freshest and it's coming in. So this tends to happen also during the neap tides. We don't get rid of that surface freshness signal, and so the strongest inflows are also at the neap tides. And then the exit out of the system is primarily sort of at mid-depth. And I think Steve hinted at this. We really have this sort of three-layered or multi-layered system where you have fresh water inflowing at the surface, deep water coming in along the bottom, and then probably exiting water on a mean state going out at mid-depth. All of these arrows are in some sense modulated every flood and ebb. So the entire water column is sort of moving in during the flood and moving out during the ebb, but generally the surface water comes in a little bit more, the mid water goes out a little bit more on the ebbs, and the deep water coming in. Yeah, Frank?
Yeah, just to add a little bit, on the spring side, you do have surface outflow.
You actually get surface outflow, okay.
Strong surface outflow, that's sort of the
Okay.
shoot out the inlet.
Right.
So the flow at the surface is not always in, it is bidirectional.
Okay. So the other, well, Frank sort of brought that to my memory is he mentioned something about, we're talking about these deep water renewal events and the salt coming in during the upcoming season. The system right now, and Rich and I have talked about, Rich Powell at UBC, about how perhaps sensitive, we look at it, we think it may be very robust, but how sensitive the whole system is to this coupling of strong estuary flow driven by the Fraser River freshet right when the upwelling season is happening. So we saw that plot earlier. I don't know who showed it, that the upwelling season, it was Roberta perhaps, so it showed the upwelling and the Fraser discharge more or less in sync. And Frank hinted at the fact that we could see climatic shifts in that. We could see the Fraser River discharge happening earlier in the year, or maybe even reduced if there's less snowpack. We could actually see the upwelling season shift as well. And I'm not sure whether the forecasts are for earlier or later in the season. If those two processes become decoupled in time, then the estuarine circulation and the upwelling, if they're not in sync, we wouldn't necessarily see the same amount of salt coming into Saanich Inlet or into the inland basins, because you need both of those to happen. Estuarine circulation driven by the Fraser River is really what's driving the exchange. The upwelling is bringing the very salty water at the entrance to Juanitica Strait. So you need both of those to get the deep water, the deepest, densest water coming into the deep inlets. Is there anything else I want to say here? Again, at any one time when you have the deep water coming in or mixed water coming in, it's coming in at multiple, it's stratified coming across here. And so it's going to come into the basin and find its density surface along perhaps the west side of the inlet and settle out at its density interface that it's going to be, that its density will determine. So the deepest water coming in, intermediate mixing. So you do in fact have renewal events happening at different times of the year, all the year long. It's not just, it's the deepest water, yes, happens during the September-October period at the end of the upwelling season when we've got the highest salinities coming in. See if there's anything else I wanted to talk about. So coming back to this 10-year time series, this again we've heard this several times. The location of this oxygen center is on the sidewall, and it's at 100 meters depth. It's actually at a sort of an interesting place for interpretation. The deep water comes in, displaces the low oxygen water past our sensor. So when we have deep water renewal, the density is going up, the oxygen is going down, density is going up, oxygen going down, density is going up, oxygen is going down. That's because the low oxygen water is being displaced upwards past our sensor. So it's a little bit of interpretation. You look at this and you say, okay, well, are we seeing deep water renewal events bringing in oxygen Yes, you are. At the same time, this is the summer period when you're respiring and consuming the oxygen due to respiration. So I'm sort of going to leave it there, but I think there was one more thing I wanted to say about circulation. It's in my mind now. And I think that's all I really wanted to say. This data, to get into the next discussion, I had this idea about an atlas. I think I talked to Steve Hallam and Mesato about our desire now, and we talked about it, which we need to put into action, publishing some data sets. And here's a really simple one we think we should publish: a 10-year time series for Saanich Inlet, where anyone can come and easily get this data in a number of different formats, hourly data, and it's published, so you actually have a reference for that data set, and you get it in the standard format, and everyone can start using it, and it's the same product. We could start doing that with the 10 years of the Echo Sounders cube that Mesato showed. We could start doing it with maybe 7 or 8 years of the Stephen Hallam's data from the genomic work in Saanich Inlet. If you have other longer time series, maybe they don't even have to be a long time series. Maybe we want to take 1 or 2 years at a time. This idea of starting to each publish or collaboratively publish our data sets, and then we can actually use them as packets and also get additional references by sharing the data through referenced data, published data sets. The atlas I was talking about could have those as a key component, and that would maybe encourage us to publish the data sets. The links in the atlas could be DOIs, digital object identifiers, to publish data sets. So I think that's it. I don't think there's any more. That's it. And are there any questions? Kim?
Yeah, Richard, can you just go back to your next before last slide?
Yes.
Okay, just clarify something for someone who's not a physical oceanographer, because it seems almost counterintuitive that the deepest mixing happens when the tidal currents are weakest. But my understanding from your talk and others is really the role of the neap tides here is to limit the freshening of the denser salty water that's coming in, so that we're getting in and out exchange at all times, and when we have neap tides, what's coming in is denser because it gets what's mixed. Is that correct?
Absolutely, and thank you for bringing the question up, because you've reminded me of the one thing I did want to say. This is an interesting little trick, and it's one of these things that for some of us it seems obvious, but it's not necessarily immediately obvious, and it can be very useful as a trick. So I'll look this way. Okay, so density. So this is our density profile in the ocean. Here's the surface of the ocean, and here's the bottom of the ocean. When we mix, we do this. We make it vertical. Now it's interesting, the surface water, which is light, gets denser, and the bottom water, which is dense, gets lighter. So when you mix vigorously, it can go like this into Harrow Strait. When you have weak mixing, you're getting the densest possible water at the bottom and the lightest possible water at the surface. So when you turn off the mixing during neap tides, that's when you have maximum stratification, and you have the least density at the surface and maximum density at the bottom, and it's that maximum density which sinks to the bottom of Saanich Inlet. So that's why neap tides give you the dense water renewal. And spring tides, you are lightening this dense water and you're densifying the surface water. Now it's interesting enough, even if you go into the middle of the ocean anywhere, any mixing at the surface always makes it lighter. Any mixing at the bottom always makes it, sorry, you got that wrong? Thank you. Any mixing at the surface makes that water denser. Any mixing near the bottom makes that water lighter, because there's no water below the bottom and there's no water above the surface. Okay, that's an interesting point. It's related to something when we all go out in these local waters, you come up to a tide line. And I get asked by sailors all the time, what's happening around a tide line? So at a tide line, you have the meeting of two water masses, typically of different density. And on the side of the tide line where you've had more mixing, it's denser. And on the side of the tide line where you've had less mixing, it's lighter. And the dense water flows underneath the light water,
and all the flotsam on the surface collects along that tide line. So if you're out boating anywhere and you look and you say, well, active passes over there, it's very vigorous, and over here there's some calm water, it's very likely the water that's come from the mixing area is going to be the dense water at that tidal point. So when you come up to a tide line, if you look around, you might be able to determine, yeah, that's going to be the mixing part of the water column. That side of the tide front is denser. And this side has had less mixing, that side's always going to be lighter. So that's an interesting perspective. The other one Anne brought up is this idea that you have these, at the bottom, these huge density gradients very near the bottom. That's a very interesting, and it's always, again, this idea that when you have a lack of mix, where you have some mixing, you make the water a little bit lighter, and where you have less mixing, you get these very strong density gradients right at the bottom. It's a little rule of thumb that's kind of obvious, but it's kind of fun. Yes, Roberta. Did that help answer? Sorry, to be fair, did that help answer? More and more so.
You were saying that you thought the Fraser River water was important to allowing water renewals to occur. I guess I would have said the opposite, that the reason we don't have deep water renewals in the early and mid-summer is because there's too much fresh water in the system. Even when mixing is low, there's still too much fresh water getting.
If you cut off the Fraser River, you cut off the estuarine circulation, and you don't bring in the salt water from the Pacific Ocean.
Yeah,
So the upwelling is happening on the continental shelf, and we never see salty water come in. We will see maybe some resident salt water. It's the estuarine circulation which pumps water out at the Wanakeuga Strait on the order of, what is it, magnifies a factor of 10. So by the time it gets into the entrance of Wanakeuga, you've got 10 times the Fraser River volume going out, 9 times the volume of the Fraser River coming in, right? So you shut that off, that water's not coming.
Because it's also what conditions with the water. When the driving force, that's circulation is pushing harder whenever the Fraser River is high in the middle of May or whatever. And then you have different water not affected you after this long down on this. And then you start getting up on the map, it's very denser one.
I think if you look at there's a time lag. I think the saltiest water doesn't get up on the shelf until
July, August is the maximum outflow. July, August is when you outflow the densest water
on the shelf. So it's a lagging.
You need that too.
You know what would be really great for this analyst is for you guys to actually explain the rest of this how this works.
Yeah, okay. Well, we're trying. We're trying to figure it out at the same time. Yes. I hope I got it.
Basically, there's been some other bits that connect little pages bringing out there.
I hope I got most of it right. I mean, I think it's almost all in your paper. So.
Yeah, I mean, that's why you got the surface going in all the time. That's not the way it is. It goes out very vigorously at the surface and in over most of the.
That's what Steve showed that, and I don't have the outgoing arrow, right? So when it's actually.
Actually, I think I missed the top 10 years. You're talking very simple. Are you talking what measurements were there? How many times did it happen?
The drifters were drilled shallow, I don't know, for 2 meters, so they had a number. 5 to 10 meters scooted out.
Which it goes out, the other stuff comes in, and everything that the whole percentage in it that goes out.
Right.
And renews the nutrients, and then you get microgrowth. So, and you can predict that roughly, given the timing, I mean, the whole annual cycle plus the spring heat things. Given the timing of the Fraser River freshet, the timing of the maximum density out in Harrow Strait, and a on-off switch on mixing based on essentially tidal velocity. Right. And so, so I mean, the pieces are all there.
Right.
But then that basically is the upper 2 layers that Steve was talking about. Because that will not get you the bottom water. Bottom water is the process you described, and is when it's absolutely lowest low mixing, and that densest water sneaks in.
Yes. Now, Deanne and Rick actually talk about the previous need being a preconditioning. I'm a little bit skeptical of that. I think sometimes the intermediate spring tide really washes everything out. But you see also the same, perhaps not quite the wonderful steps that Roberta showed, that once a month we see, somebody showed, maybe it was Frank showed, the deep water in Strait of Georgia have these beautiful spikes. Those big spikes are the dense water flowing past are, again, same sort of thing. We're now at 300 meters, but in the Strait of Georgia is 400 meters deep. So the same thing, the deep water is pulsing over on a flood tide during the neaps, flowing down the Thalweg, passing our sensors, so you get a spike in density, and it settles down right at 400 meters. So you get those spikes, and they're above the mean density here, so you know that water is residing down here in the Strait of Georgia as well. So I agree with Frank also that what's we tend to see in saanich inlet is also very similar to what's happening in the Strait of Georgia. They're same processes happening very much at the same time. Kim.
Do we know anything about transit times, outgoing, water mass construction, and rival, Victoria Silver?
I did know those numbers.
We have to look at. That's the more right. We should be
I was going to say the mean flows at that time are about half a knot, so we can do there's 7 kilometers a day, so we could do that.
For basic flushing, probably one of the essential features is the weakest tide of the year, which is in August. So that's a part of the
yes.
The basic flushing is you just have the very weakest period of mixing. And that carries into September.
It does, yes.
Yeah. So I think if you brought dense water in June, there still would be enough mixing across the basic.
I think Roberta showed that the active past tide, you had one very weak leap happened in May, and it was enough that you got a renewal. Then for the next 2 months, there wasn't a really weak leap. And then the next one in August, we saw it again. And it's again, talking to Rich, how sensitive perhaps the whole system is to, that it has to be just weak enough to, and then like I said, you really shut down the mixing, and the water that's coming in is very, or it's really got its original source characteristics. It hasn't been done.
That's the Anderson and DeVol paper, where they saw boluses, where they anticipated the bolus of water coming.
Yes, that's what we see in the Strait of Georgia with these pulses. You can trace them back to exactly during a flood tide, and it almost gets during the air that gets held at the boundary pass sill, and then burps over during the next flood tide, and you get a bolus that comes all the way running down in the Strait of Georgia.
We saw that in our time series data too. There was one renewal where we sampled quite frequently, and you would see one time you go sample, there's some oxygen in the water, the next week you go, there's no oxygen. The next week after that, the oxygen goes again.
So that brings up an interesting point, and I know the UBC team goes out and they're saying, okay, we go in every 2 weeks, and it's exactly the same time of the tide. I think it's very important, and I'm going to be hedging my own bets to say it's important to know where you're going out relative to the tides, and depending on what you're looking for, what part of the fortnightly cycle it matters.
Absolutely.
It absolutely matters.
We really try to plan for it.
And the beauty of tides is you can set your clock by it, you can plan it a year ahead.
Yeah, but there's a monkey wrench in there, and that is that the same tide will not always necessarily bring in bottom water. Depends on what's the actual stratification.
Yes,
exactly. So
that's
when
there's no renewal about the high.
Yeah. So. Yeah.
It's very warm water from the ocean.
Yeah, that's right. So there's you get two things. Yes, you need to know the tidal phase, but you also need some information about what's the density difference outside.
Do we have something like that? Is that something that you know?
Well, we don't have any, again, what you want is a vertical profile in Harrow Strait or Victoria Sill, and we've got some bottom, and getting the water, the profile coming in would be very desirable. We almost have nothing except for way up in the shelf, all the way to saanich. It's just, we have some bottom moorings, but not real time. And getting real time mooring type of profiler and strong currents is just a technical challenge.
One convenient thing is that the line features and the operating crews tend to leave right around the time you will allow us in September, and they do stop and turn us ready. So that.
But as we saw today, or with Frank's, there was literally a vertical profile from Harrow Strait, about a degree warmer one year from the next. But I think we also pointed out that in fact, the same day you might find a different, I know Rich and I were up there in the late 90s, and we had a Tully cruise, and it was the vector. We were out in Harrow Strait, and we had a CTD, and we got caught, it was on the vector, we got the ship caught in a boil, so we were in 300 meters of water, and the classic boil signature, the water's coming up, and it's sort of spilling over the surface. You have this massive boil, bigger than the size of the ship. And we were drifting through in the vector, and the Doppler that we had up the side was measuring the entire 300 meters of water. And I was right to the bottom as far as we could tell, upward currents of 40 to 50 centimeters per second in this boil. And we did a CTD cast, and it's absolutely uniform. Everything was absolutely uniform for 300 meters. And we sort of drifted around, and 1 or 2 hours later, the tide had calmed out, we couldn't see any boils, and we started to do CTD casts to find that density profile, and it had obviously pancaked out and gone, and it was really remarkable. You do it 2 hours later, and everything stratified again. That doesn't make sense. But it must have pancaked out. That whole water column must have shifted and pancaked out somewhere. But it's, so measuring in Harrow Strait is a challenge because it's very curious by when you're has anyone had any comments on this idea of an atlas? I think it's a big community effort. It takes a lot of contributions. It could have lots of different layers, geology, history. It could go terrestrial, but an atlas that has a really comprehensive view of what's known about saanich inlet and what's happening and the data sets that are available. Does this sound like something the community would want to say, yeah, let's keep talking about that, let's start that?
I'll just speaking to Helen, and then we actually are going to run. But we've been putting together two papers for scientific data, which features peer-reviewed data publication journal. So really high-quality data sets, not necessarily time series, but definitely the time series is something we have a lot of in saanich, so it's great. Monica's been championing all of our clinical data and working on the molecular biological side, so there'll be two kind of companion papers, but not that difficult to put together, especially given, that we already have a lot of baseline here. We already have, we're talking to each other. You don't want to get too many different data types into one paper because the review becomes quite challenging, which is something we've run into in trying to put our data sets together. But that being said, I think we'd be happy to circulate our draft. People want to see what that looks like. But there's a note on quality control and looking at methodology, absolutely no analysis other than quality control. So that makes it, even if you have data sets that you might not ever publish, but the data is sitting around and you create to communicate, community, maybe even a citation. Definitely.
Same thing with this 10-year data set, just these 4 variables. Now, this is available today, right now, for free for everyone, but how do you reference it? You can't reference it. So the simple task of writing up 2 pages that says, these are the measurements that were made, here's how it's been processed, here's what it is, and here's the link to it. Now we can reference it and everyone can use it.
We want people to use our data.
Yep.
We want to afford the scientific community, or people we work directly with, and anywhere, but it's always great when you get it.
All right, so to help stimulate this, we will go away from this workshop and we will continue to develop what we're calling our saanich inlet working group. It'll have a web page eventually. We'll ask you to register to be a participant in that, and you can either join passively and just listen to what's going on. Anyone can come visit the material on it, but if you're registered, then you can actually be posting material and participating in the discussions, and it'll be a place to at least a place for gathering, and then we can figure out where this atlas idea might go. Okay. Roberta's concentrated efforts starting in September. The Ocean Observatory will keep the buoy profiler going, the autonomous sail going, the VIP going, maybe get the glider in the water. I think the gliders it's going to need to go across the inlet as well as up and down. Anyone else who is thinking that they want to participate to add the richness to what we may be collecting in the next year, contact Roberta. Rena, did you?
Closing remarks. Saanich Inlet is an incredible place. Sold my house and we bought on Saanich Inlet.
I just wanted to look at it. And it's also beautiful. And it's been 10 years of data collection, but it's been 15 years of preparation to get there. I went around this room and I realized Jim was there, and Frank was there, and Ann was there. Brian, you might have been there, no? And Dave was there when we were doing the initial planning back in the year 2000. At the same time, Chris Barnes was catching as well. It's been a journey. Richard and I wrote the grant proposal and funded in early 2002. And we hired Adrian around that year. Right after he was hired, he came to me and he said, okay, where do you go? Where are the engineering plans? Drawings. Where are the engineering drawings? So I took out the grant proposal. I said, no, there are not drawings. There aren't even specifications, science specifications. There's a lot of wish lists here for the science. We started off from there, and at the same time, there was a group of colleagues in the United States who wrote a grant proposal to NSF to join onto the Juan de Fuca line. That's right. Remember? We were going to transit all across Juan de Fuca straight going out from Ray's office over. When it was reviewed in NSF, the response was, those Canadians will never be able to do step one. Not fun. So we took the money from that line, poured it into the rest, and sent it to the I think it's such a hugely rich environment. It was, for me, a fabulous 12 years of my life on that ride, probably some highs and extreme lows as we went along the line with the most fantastic team, Richard, Adrian, and another group, and the rest of the group, and then matching through as Chris built up this group. And so it's just wonderful to see where we're going today with the second proposal. Well, I wanted to thank you, Richard. I wanted to thank you, Akash, for putting all this together, and Mike as well for your persistence here and Karen on the side. So it's been a great time, and thank you very much.
You're very welcome. And we'll plan another one in 4 or 5 years. So this needs to be an ongoing saga, and I think it was very encouraging to see old familiar faces and new ones with the graduate students. But one thing we were missing perhaps a little bit are some new principal investigator faces in the room. So that's something I think we need to elevate. The graduate students in Maysado, I'm going to use you as a good example. Very interesting case where somebody comes and builds their thesis and starts building their career working in observatory data, takes that experience somewhere else, another institution, the University of Washington, world caliber facility, then goes on to Oregon State, working with some remarkable people, and they hire her because of that experience with the observatory. And she's now going to be looking at linking cross-observatory observations. So seeding graduate students around the world is something we could sort of benefit from. And so I encourage you graduate students, please contact us, get involved, start using the data, but use the data for the rest of your career. It would be helpful for us too. Any other closing remarks by anyone, or we'll wrap it up there? Akash and I will put in the queue to try to get out a preliminary part of a sort of a workshop draft report. We will be contacting all of those who make presentations to see if they want to send us a PDF version, culled or corrected, to include in the proceedings. And we'll put in summary notes. We'll have on the archive the recording. It probably won't go on the website as is, but it'll be there for reference. And we'll get the saanich and the working group page up and running, and this will be one of the reports that will be on there. Anyway, good. Thank you very much. Is this the...